So good morning, everyone. It is so exciting to host this conference today. And I'm really thrilled to be able to open up the BIRAX UK Israel Healthy Aging webinar. Now, I think that many of you know that we were supposed to have this meeting in person. So allow me to introduce myself. I'm Karen Abraham. Among other things, I'm the Vice Dean at the Faculty of Medicine at Tel Aviv University. And I've taken it upon myself, along with a really fantastic team, to organize this conference, which is probably going to turn out to be a series, since this is the first rendition. And I very much hope that we'll be able to host many of you, if not all of you, in Tel Aviv in June. The original dates were supposed to be today and tomorrow. And there was a bit of a question whether we should move to an online version. We've decided to go ahead. Fortunately or unfortunately, we're still aging, some for the better, as you know. Times there are improvements with age, but also because the science is continuing, and it's important that we hear each other and we get to have a discussion and talk about this really quite overwhelming topic from so many different aspects. So first, I want to give a couple of acknowledgements because I've had so much help in organizing this conference, the original one, the replaced one, and then certainly the one in June. So first and foremost, I want to acknowledge BIRAX and the British Council and the UK Science and Innovation Network for their financial support, which is making all the difference to being able to host all of you in Tel Aviv, hopefully in June. I also want to acknowledge Iris Barner, Jonathan Mandelin Shore, and Dennis Patchuk for all their help in getting this meeting going. My co-chairs, Dame Linda Partridge, Peter St. George Hislop, and Daniel Barr, as well as the Israeli Academic Committee, Ehud Cohen, who we're going to hear from, Ilana Gozes as well, Mira Marcus Kalish, who will be chairing tomorrow, and Dov Shmotkin. I also want to acknowledge our UK Academic Committee, Sarah Harper, who we're going to hear from tomorrow, Karen Steele, who's chairing a session today, as well as Richard Sell, who will be chairing as well. So without further ado, because there's a lot to say and a lot of people to hear from, let me just give you a couple of rules. I am going to keep everyone on mute so that we don't hear background noise. We have a great number of people who are registered. When it comes time to questions, please raise your hand under the participants where you can raise your hand. The chair will actually see that in the column on the right and will call on you, and then you'll have the opportunity to unmute yourself and ask the question. So let me start by introducing Neil Wiggin, the ambassador. And do we have you here? Where can we find you, ambassador?
Hi, yes, I'm here.
You're here. Okay, so we'll put you on speaker view. Wonderful. So we are so honored that you've been able to join us. So I think that many of you know that Neil Wiggin is the British ambassador to Israel. He was appointed in 2019. He's a University of Oxford graduate, as some of you are coming from Oxford now, so it gives you a lot to be proud of. And he was appointed Officer of the Order of the British Empire, as well as many other accolades over the years. So we're really very honored, Ambassador, that you've decided to join us and to give us some greetings, please.
So thank you very much, Professor Abraham, for that introduction. And as you said, we had planned for this to be a full in-person academic conference. Clearly, because of COVID, that wasn't possible. So we've moved to this online format. And I'm really grateful, as you said, to all the people who worked really hard on the initial idea and then to transform it into what we're doing today. And I know a lot of work has gone into that from my team and elsewhere. So I'm really grateful that we can do that. And I think it was absolutely right that we did it because Corona has only shown the importance of the aging agenda. It's clearly a disease that has struck aging populations most seriously, as we all know. So we thought it was absolutely right to keep up the momentum which we have developed in our work through BIRAX and other endeavours around aging. And we also think that the agenda of the webinar is very appropriate. It's very multidisciplinary. We've got presentations on neuroscience, on Alzheimer's, on cognition, calorie restriction, heart disease, precision medicine, and many other issues. And also it's looking at the social aspects of aging, such as inequality and aging in the UK and aging in the workplace. And of course, we wanted to carry on because BIRAX for us is a flagship part of cooperation between Israel and the UK. That's something that the ministers in the UK really want to make a push on. We see a huge potential for collaboration between Israel and the UK in cutting edge science and cutting edge technology and innovation. We think Corona has really emphasized the importance of international collaboration, and we've worked really closely with the government on developing a response to Corona. So through BIRAX and other initiatives, we want to keep that work going forward. As you know, BIRAX Aging has so far funded 7 collaborative research projects with a value of £2.8 million. We're developing a call for proposals with our partners, and we're keeping on working with the theme of healthy aging. We want to put a big emphasis on the development of early career research through our funding opportunities and engagement, as we think this is something that will really help develop UK-Israel collaboration through the decades, really. Following this webinar, we plan to do some online events for early career researchers. We will put out the next call for proposals in the first quarter of 2021. And as Professor Avram said, we'll hold the BIRAX conference in June 2021. I did want to say some thank yous, so particularly to Tel Aviv University, who've worked so hard with us on this conference, to the conference academic chairs, Professor Avram, Professor Daniel Barr in Israel, and Dame Linda Partridge and Professor St. George Hislop in the UK, and to, as you said, to all the academic committee members in Israel and the UK who've worked to put this together. And on behalf of the embassy and the British government, I really want to thank our partners who've provided us with support through this time of uncertainty and been really positive about us showing flexibility and moving to respond. Particularly the PAIRS Foundation, which has been BIRAX's founding partner, has been with us all the way through and has given us support through this crisis. And also to the Israeli Ministry of Science and Technology, Parasol Foundation Trust, the Rose Trees Trust, the Charles Wolfson Charitable Trust, the MS Society, the British Heart Foundation, and Diabetes UK. So a big thank you from me, from my government, and from everybody at the embassy and the British Council for all of that. With that, I will now honour Karen's demand for us everybody to keep to time and hand over to the main event and the rest of the webinar. But I really hope this will be a useful and productive webinar, and I'm really grateful to all the presenters, but also to the people who get time this period. Thank you very much indeed.
Thank you so much, Ambassador. That was really heartwarming. A lot of organizations and people are involved in making this all happen. So let me move on to our next, really, I'm just so honored to introduce Dame Shirley Porter. And I think that many of you know Shirley because she's what we call in Israel Ben Bait, or it would be more correct to say Bat Bait. She's part of our house here at Tel Aviv University. So Dame Shirley Porter is, I think the way to really be able to describe her is as a visionary. She's the former Lord Mayor of Westminster, London. Probably many at Tel Aviv don't know that about her. But what really she's known for, certainly in among our cohort, is all of the work that she's done for the environment. In fact, she's gotten the Green Globe Award for that honor. And she's very much involved in supporting the arts, education, and health issues. And when it comes to aging and healthy aging, she's always here supporting us with her absolute enthusiasm and just wonderful, warm words. So Shirley, please, if I can give you the stage, and let's hear a few words from you.
Thank you, Karen, and greetings to you all. I normally prefer to speak off the cuff, but like a lot of older people, including President Trump, and the man likely to become president 3 weeks from now, Joe Biden, my ability to stay on message has decreased with age. So I'm going to do everyone a favor and use notes I prepared, which I hope won't be too distracting. Firstly, let me say how pleased I am to be again included in the BIRAX conference and how honored I feel in the company of this year's distinguished experts on aging, courtesy of Zoom, which is truly a blessing during these times of Corona. During most of my lifetime, I couldn't have imagined today's amazing technology, let alone using it to connect and communicate. I'm also grateful to be able to participate this morning. The Corona pandemic has dramatically altered the lives of everyone, especially people of a certain age, and life has become alarmingly unpredictable. Whilst I'm more fortunate than many others who sadly live in loneliness and fear, I will be 90 at the end of next month. Therefore, I qualify as one of the endangered species with all its ramifications. Thankfully, so far so good, and I'll keep wearing my mask, keep my distance, wash my hands, exercising, trying to make sense of the conflicting medical advice, and taking vitamin D, C, and zinc. In times of public emergency, social truths are revealed. The coronavirus is one such emergency, and to quote Shira Shimoni, a PhD candidate at King's College London, it reveals that the lives of the elderly appear to matter less and in some cases are even deemed disposable. The reasoning apparently was that since we were going to die soon anyway, emergency health services should be allocated to younger people. Thankfully, the thinking on that seems to have changed somewhat, but I myself, I'm very happy not to have had to take part in the conversation of who should die and who should live. People are living much longer and in many cases still contributing to the world. I just read an interview with my pin-up boy, Sir David Attenborough, aged 94, in which he talked about his latest documentary. He says he's lived far longer than he ever expected. And I say, thank heavens. Look, not only at all he's done, but what he continues to do. Last night, I watched his documentary and I really was in awe. He is elderly, yes, but far from dispensable. And he recently opened an Instagram account to make his case for protecting our planet with younger audiences. He's the world's most revered and listened to naturalist because after more than 70 years studying biodiversity and the environment and emphasizing the necessity of striking a balance in the world, Sir David knows what must be done. Heading into my 10th decade, I wondered what pearl of wisdom I might share. I decided as a former Girl Guide, I'd say, be more prepared. This isn't the first global crisis, and it won't be the last. But we always seem to be starting from scratch, as if we learned nothing. To paraphrase George Santayana, those who cannot learn from the past are doomed to repeat it. I don't understand why knowledge and awareness gained during a disaster is not studied in depth and applied to preventing or combating the inevitable next crisis. Instead, there's the tendency to crisis manage. Help, help, crisis. When disaster strikes, handle it and set it aside until inevitably something else comes along to wreak more havoc. I want to refer to wildfires at this moment, which are an example. Every year, Australia, Brazil, Russia, the US, and many other places lose millions of acres of land and animal life in the hundreds of thousands, yet governments and politicians are still too slow to find solutions. Finally, I'm pleased to say, the US and Australia are beginning to consult their tribal elders, Native Americans and Aboriginals, to understand the methods that they use for centuries to prevent the devastation of wildfires. I'll add that I hope that everything we're learning during the COVID crisis will be retained for future use. I'm sure that this particular forum will. Also, to my mind, it makes good sense to deal with most issues locally. Perhaps I'm biased because I was a leader of Westminster Council for many years. Experience shows that a crisis can be handled more efficiently and effectively using local councils and their infrastructure, and local volunteers who are well prepared to take on any emergency. I suggest that the motto today should be thinking small and efficient rather than big and bureaucratic and inefficient. And I have the feeling get a lot more done. Before concluding, I would like to pay tribute to Shlomo Suleiman, not only Israel's oldest man, but the world's oldest, who passed away on Sunday, aged 117. Until the panic, he was healthy and active and lived independently. It appears that his motto was exercise and don't eat too much. This might be difficult to follow, but I understand where he's going. His family attributes being forced to live in isolation during the coronavirus to the deterioration of his health. He died now, his family said, because of the solitude and loneliness of the pandemic. Solitude and loneliness, definitely something for the experts to think about. Now I'll hand over to Karen and settle in what I know will be a very interesting conference. Stay well, and thank you.
Shirley, thank you so much. Such wise words. Wonderful. So next, I just want to give an acknowledgement. Professor Ruth Arnon is here in the audience, and I think many of you know that she was the president of the Israel Academy of Sciences and Humanities and is in the UK Israel Science Council and has contributed an enormous amount. There are just not enough words to describe how much Ruth has contributed to both Israeli science and science worldwide. So thank you, Ruth. It's really an honor that you've joined us today. I also want to follow up on something the ambassador said regarding the grants that have been given for aging research by BIRAX. So we do have a couple of those partners who are going to be speaking today and tomorrow. So our next in line is Professor Karen Steele. And Karen is a dear friend. We've worked together for many years. She works on hereditary hearing loss, and she's now working primarily focusing on progressive hearing loss, age-related hearing loss at King's College London. She's also been awarded, among other things, the Brain Prize. So Karen, please take over and you can start the next session. Thank you.
Good, well, welcome everybody. And thank you very much for that introduction, Karen. I'd like to start by calling on Robin Franklin to give the first talk this morning. Robin is based at the University of Cambridge in the Stem Cell Institute there, and he's Professor of Stem Cell Medicine at the Clinical School there, the University Clinical School. And he's an internationally recognised expert in remyelination biology. He received a prize in 2017, the BIRAX International Prize for Research Innovation, and he's also been elected as a Fellow of the UK Academy of Medical Sciences. And so I'd like to call on Robin to give the first talk, which is on regenerative capacity of adult CNS progenitor cells. Over to you, Robin.
Thank you, Karen. I've got very unstable internet this morning for some perverse reason, but I'll press on and see how we do. Can I just check that you can see my screen?
Yeah, we can
see it. Fine. Okay, great. So in this talk, I'm going to describe some background to work that I'm going to be doing over the next few years in collaboration with my friends and colleagues Kevin Shalou at the University of Cambridge and Amnon Buxbaum at the Hebrew University of Jerusalem. So the mammalian and human central nervous system contains a variety of different cells, which is shown here. Neurons, of course, you will know about, but there are other interesting cell types. And the cell that has been the focus of my research for several decades now is the myelinating cell of the central nervous system, the oligodendrocyte. And this is a cell whose star is in the ascendancy, and that's for two reasons. Firstly, because in contrast to pathologies of neurons, if you lose oligodendrocytes, there's a spontaneous regenerative process whereby you make new oligodendrocytes that make new myelin sheaths. And this is the process of remyelination. So oligodendrocyte loss is followed by a spontaneous regenerative process in sharp contrast to the loss of neurons, which are not replaced. And the second reason oligodendrocytes are in the ascendancy in the neurosciences is because it turns out that during adult life, there is constant myelination taking place. So this is a slide I borrowed from a review from Heidi Johansson-Berg, who's speaking later in this meeting. And there are, in the adult brain, there are exposed areas of axons which can be myelinated. And that, of course, changes the conduction properties of the nerve fibers and therefore the functioning of the circuit. So this adaptive myelination, as it is called, underpins the acquisition of new locomotor skills. And there's increasing evidence that it underpins the acquisition of cognitive, that it's involved in cognitive function. The source of new oligodendrocytes in the mammalian and human brain is a very widespread population of multipotent progenitor cells, or stem cells. Whether they're progenitors or stem cells, you can argue the toss. I'll call them progenitor cells for the purposes of this talk. So all of the yellow and red dots that you see here in this mouse brain are this population of progenitor cells. So you see that the brain is actually full of a stem cell population. There's no shortage of them. They're very widely spread. And so what happens if you need to make oligodendrocytes, particularly in the context of a demyelinating disease such as MS, is this population of progenitor cells becomes activated. They migrate and divide like bilio, and then eventually they'll differentiate into new oligodendrocytes to make new myelin sheaths. So that's how remyelination takes place. And the basis of adaptive myelination is not dissimilar. The problem, of course, is that as you progress throughout adult life, like all regenerative processes, remyelination becomes less efficient in a progressive way. And the failure of remyelination in chronic demyelinating disease such as MS results in irreversible loss of neurons and axons, and that's why the disease becomes progressive. So we're interested in this from the perspective of neurobiology, but there's also a translational opportunity here to make sure that you go down this pathway invariably and not down the degenerative pathway. So we've hypothesized that with aging, something takes place with the progenitor cell. The progenitor cell goes awry, and that has knock-on consequences for the whole of the process by which oligodendrocytes are generated. It turns out actually the number of your progenitor cells doesn't change with age. The number you have in your 20s is the same as the number that you have when you die. But their properties change profoundly. So a number of years ago, we cracked a long-standing problem in the field of being able to isolate progenitors from any age of laboratory animal. And if you grow progenitor cells from aged rodents, it transpires that they have all of the hallmarks that you associate with aging stem cells. So they acquire increased DNA damage, they increase senescence markers, and most profoundly, they fail to become oligodendrocytes as efficiently. So the red lines is the production of oligodendrocytes from young adult progenitor cells. The blue line is the severely impaired production of oligodendrocytes from aging progenitor cells. Now, it turns out that actually the properties of the aging progenitor cell are not set in stone. They're not immutable. You can do things to galvanize or rejuvenate the properties of the aging progenitor cell. So a number of years ago, we used the model of heterochronic parabiosis to show that if you bathe an old progenitor cell in serum from a young animal, you would perk up the aging progenitor cells so that it worked efficiently. Last year, we published a paper where we showed that calorie restriction in aged animals is a very profound enhancer of the properties of the aging progenitor cell. And you could also phenocopy that effect using a calorie mimetic drug, metformin, which some of you will be familiar with. It's the frontline treatment for type 2 diabetes. And in fact, the enhancement with both calorie restriction and metformin of failing regeneration of myelin in the aging central nervous system is extraordinarily profound. These are non-overlapping data sets. It's the most impressive enhancement of failing regeneration in the aging brain that I think has been published to date. Now, a question that we've asked, and we've done this, this is a collaboration with a physicist colleague of mine, Kevin Shalhoub, was done by an extraordinarily able PhD student that we co-supervised, Mikey Siegel. The question we asked is, why does the stem cell, the progenitor cell in the brain, lose function as a consequence of aging? There are two possibilities. Either there's an internal or intrinsic aging process within this population, or the progenitor cell simply reflects the age of the environment in which it exists. So in the aged brain, the progenitor cell neither proliferates nor differentiates with any great efficiency. So we argued that if there are intrinsic changes in the aging progenitor cell, if you were to put them into an environment where proliferation and differentiation occurs very efficiently, that is to say the developing central nervous system, they should continue to proliferate and differentiate poorly. In fact, the exact opposite happens. If you take these functionally impaired aged progenitor cells and put them in a developing central nervous system, they proliferate and they differentiate with the same efficiency as a young stem cell, young progenitor cell. So that tells you that the change in function within this progenitor population is not locked in and is more reflecting the environment in which it exists rather than the chronological aging of the progenitor cell. Now, to cut a very long story short, we tried to find out what it was that changes in the environment which makes the stem cell function less efficiently, the progenitor cell function less efficiently. And using the technique of atomic force microscopy, we showed that the aging mammalian brain increases in stiffness quite dramatically. That's what this heat map on the right shows. So the young rodent brain has the stiffness of a ripe avocado, whereas the aged rodent brain has the stiffness of an unripe avocado. So there's an appreciable change in just the physical properties of the aging brain. Now, we wondered whether actually that physical change per se was sufficient to instruct the progenitor cell to behave either as a young in a soft environment or an old progenitor cell in a stiff environment. And what we did was to take acrylamide gels, which are biochemically inert, but were tuned to the stiffness of a young or an old brain. And if you take an aged progenitor cell and put it on an acrylamide hydrogel substrate that is tuned to the stiffness of an old brain, it fails to proliferate and it fails to differentiate. But if you put an aged progenitor cell on a soft substrate with the substance, with the stiffness of the soft brain, it kicks into cell, proliferates, and more significantly, it undergoes differentiation. And that's shown graphically here. And these are not just changes in these two properties, these are holistic changes in the progenitor cell. So the entire transcriptome changes from one of an aged progenitor cell to a young progenitor cell. The age state of these cells is extraordinarily flexible. So that's very interesting. So what could you do to make the aged brain work more efficiently? Could you decrease its stiffness? Well, it transpires that that's an extraordinarily difficult thing to do, and we weren't able to come up with a safe and sensible way of doing it. But Mikey Siegel had the bright idea of, instead of changing the environment, you change the ability of the progenitor cell to respond to the physical properties of its environment. And we searched through many ways in which that might happen and hit upon a mechanosensing molecule called piezo-1. So this is a molecule which enables a cell to register the physical properties of its environment. And if you take piezo-1 out of aged progenitor cells, they no longer know that they're a stiff environment. So if you do the experiment I showed you previously on the hydrogels, and you take an aged progenitor cell where you've deleted piezo-1, it doesn't realize it's on a stiff environment, so it proliferates, and what's more, it differentiates more efficiently. The final experiment we did in this study was to show that we could also do this in vivo. So what we did was to develop AAVs, which we delivered systemically, and using CRISPR-Cas9 gene editing strategy, we were able to specifically delete piezo-1 in the progenitor cells. And so if you take an aged animal and induce a demyelinating injury, you remove piezo-1 from the progenitor cells so they don't realize they're in an aged environment, and they proliferate more efficiently, and what's more intriguingly, they differentiate into new oligodendrocytes much more efficiently. In other words, they begin to regenerate as if they were young progenitor cells. So just to conclude, this is our model of how the aging brain changes the properties of these fundamentally important progenitor population that are involved in regeneration and, as I alluded to at the beginning of the talk, cognitive function. So with age, there's an increase in ECM production, which increases the stiffness of the brain. Through piezo-1, that changes the properties of the progenitor cell, rendering it less efficient. And so you go into this feed-forward process. So that's just by way of summary. I should just acknowledge the members of the lab who did this work, principally Bjorn Neumann, who was instrumental in many of the aging progenitor cell studies that I've described. So I hope I haven't kept my clock going, but I finished there, Karen.
Good, thank you, Robin. No, you're well in time. Thank you. We have some time for some questions. So if you click on the participants icon at the bottom, then the list of participants will come up, and there you'll be able to raise your hand. There's a little button on the bottom right if you want to ask a question. So I'd like to start asking the questions by asking, what's the link? Is there any link between the effects of metformin and diabetes type 2 and the stiffness of the brain?
Yeah, that's a key question, Karen. And we haven't done that experiment yet. For two reasons. One is that they're quite cumbersome long-term experiments to do, and I haven't had the outcome of a grant that I've written to ask to do those experiments. But that's exactly right. Whether metformin changes brain stiffness, I don't know, but I would bet my money that calorie restriction changes the physical properties of the aging brain.
Mm-hmm. Okay. If I just follow up on that, do you have any idea why the brain becomes stiffer as it gets older?
I don't know that there necessarily is a why and a wherefore. I think once you've gone past the point of being reproductively useful in a Darwinian unit of existence, I don't think functionally it really matters very much. So I think in the aging brain, there's no selection pressure for it not to go into sort of free fall of stiffening. What I think in that's fairly philosophical, and I think in mechanistic terms, what's happening is that the astrocytes, another major cell population within the brain, it becomes more pro-inflammatory with age, and that's well recognized. When it becomes more pro-inflammatory, it increases its ECM production, so production of exocytosis matrix. That increases stiffness. We now have some unpublished data that increased stiffness makes the astrocyte more pro-inflammatory. So you can see that the brain is going to this sort of vicious feed-forward cycle where there's more inflammation, more stiffness, and you're just into free fall into an environment which is not very permissive for plasticity.
Mm-hmm. Okay, that's really interesting. We've got a question from Anat Bensby. Would you like to?
Good morning, wonderful talk. I wanted to ask, what do you think, or your thought on what is PESA1 needed for to actually sense stiffness, because it seems as it's a negative thing in this process.
Yeah, well, that's a great question. So we did actually append some very preliminary data that in a supplementary 15 in that paper that sort of supports this developing notion we have that actually it's a developmentally expressed protein. So of course, when you're myelinating in the first instance, at some point you have to stop. And actually, it's quite a clever way of timing the end of myelination by increasing the stiffness of the brain. So I think it's a developmentally important mechanism, which in the aged brain becomes dysfunctional.
So it's upregulated and unregulated, basically dysregulated, I would say.
Yeah.
Okay, I have a question from Richard Show.
Robin, great talk. Thank you very much. I'm a vascular biologist interested in the brain microcirculation.
Yeah.
Does the stiffness also impact on the brain microcirculation stiffness and vasoreactivity?
Yeah, that's a great question. In fact, that's something we're just exploring right now, in fact. And I don't know the answer, but almost certainly. I mean, I think these are pan-physiological effects, and so I think there will be few exceptions of any cell type and any physiological process that isn't disturbed by this atypical non-physiological environment.
Okay, good. I think there are no more questions. Thank you very much, Robin. It was a really interesting talk and a great start to the meeting. So thank you. We'll move on to the next talk then. So can I call on Professor Ilana Gozes to give the next talk, which is entitled "A New Look at Alzheimer's Disease Diagnosis and Treatment Towards Healthy Aging." Professor Gozes is in Tel Aviv University. Hello, morning. And she's won a whole list of prizes: the Olsen Prize, Julidan Prize, Tiva Prize, Neufeld Award, Hans Wissenschaft Kolleg Fellowship, Humboldt Award, Landau Prize, and Rare Champion of Hope Award. So an enormous long list of prizes that you've won, celebrating your research. I will like to pass over to you now to give your talk. Thank you.
Okay, thank you very much. I will upload my presentation. So I would like to start by thanking Karen and all the organizers for having me here, and I'm very honored to be here. And my English is not going to be up to par, but I'll try. So I'm looking at, with a new look at Alzheimer's disease diagnosis and treatment towards healthy aging. And I also wanted to kind of preface by saying that I look at aging and I also look at development, and I think that there are some commonalities when aging goes awry and when the development goes astray, and when you have autism and developmental disorders. And the two protein hallmarks of Alzheimer's disease are deposits of amyloid beta and tau pathology, namely the microtubules, which are the building stones of the scaffold of the nerve cell, fall apart. Not only the nerve cells, also the oligodendrocytes and the astrocytes that we have just heard about. And we have deposits of the protein tau, and this is very appropriate to Tel Aviv University. And so I even edited a book called From Neuroprotection from Autism through Schizophrenia to Alzheimer's disease. So our first look was to say, can we look and find diagnostic biomarkers in blood? And for that, we used lymphoblastoid cells. And we looked at the lymphoblastoid cells. These are immortalized white blood cells. And we looked at them to see, can we distinguish between cells that are highly sensitive to amyloid beta, that's the first deposit I have described in Alzheimer's disease, or less sensitive. And we found a sensitivity like that. And we found that a protein called regulator of G protein signaling 2 is going down, and this protein senses this amyloid sensitivity and could be maybe a marker for Alzheimer's disease. So this is like a general look, and we looked at the whole transcript of all the proteins that are made by these white blood cells. Then another look was just to look at proteins that are associated. Somebody else is talking with me. Sorry. So another look at Dina Gidron, can you please close your microphone?
Everyone, and then you unmute yourself, please, okay?
I am unmuted.
Yeah, first I'm going to unmute everyone. I'm going to mute everyone, and then you unmute yourself, and then continue.
Okay, so now I can continue. So another look, so we looked generally at all the proteins. Another look is to look for specific proteins that we know are associated with longevity. For example, SIRT1 is a protein associated with longevity. And recently it also was found as protective against tauopathy. You have more SIRT1, you protect against tauopathy. What we showed is also that it goes dramatically down in Alzheimer's brains or Alzheimer's lymphoblastoid cells, and it goes up in centenarians. So maybe those that live to 117 have more SIRT1, which protects their brain. So this is something to take into consideration, and this can also be a marker, a peripheral marker. And now it's stuck. Sorry, my internet. This is stuck. Sorry. So going to my my next subject, Sorry about this. It is the connection probably is not that great. So our discovery like 20 years ago was of a new protein to science that we called activity-dependent neuroprotective protein. So this is again, like looking at sirtuin or SIRT1, we also looked at the protein that we discovered to see the connection with Alzheimer's disease. So we were looking for proteins that protect the brains and are secreted by the support cells, by astrocytes, and then protect nerve cells. And we discovered new protein to science that 20 years ago that we called activity-dependent neuroprotective protein, or Adnp in short, that protects neurons against electrical blockade, hence the name. And when we knocked Adnp out completely in mice, we didn't get mice because it is essential for the neural tube closure and the formation of the brain. We also derived a very small peptide out of Adnp that mimics the neuroprotective activity that we call NAPVSIPQ. And recently, just a few years back, a syndrome was discovered that is called now the Adnp syndrome in children, that they have de novo mutation in Adnp, and then they have intellectual disability, autism, and motor disabilities. And this is one of the most prevalent syndromes among the de novo mutations. So this occurs in pregnancy or in the germline, and the children are autistic. And here you can see among 102 ASD genes, Adnp is one of the leading ones. So if you look at the mutations on the Adnp protein, you see that there are very many with some hotspots. And you see here me with the mothers of the Adnp children, some of the mothers, and you see they have facial dysmorphism, and they also have developmental disorders in terms of motor development, as you can see here. But the most prevalent problem is intellectual disability. And the other problem that they have, which could be diagnostic, is they have early tooth eruption and the teeth are not well developed, and this also connects with aging. So the intellectual disability connects with the mild cognitive impairment that we see in the aging, and the early tooth eruption and the teeth that don't develop properly connects with losing teeth and in the elderly. And then if we look to see what happens to Adnp in the aged population, so Adnp in the serum is correlated with cognition, with the IQ tests, a complete proteomic of Alzheimer's disease serum compared to normal healthy individuals, age match. And this was not done by me, this was discovered in Taiwan. The only protein that went down in the serum was Adnp. And also, if we look to see which genes Adnp regulates, so it regulates hundreds of genes during development and also in the mature animal. One of the major genes regulated by Adnp is apolipoprotein E, with apolipoprotein E4 being the major risk factor for Alzheimer's disease. So how does it work in terms of neuroprotection? So it regulates hundreds of genes. So this is one way that it works. The other way is that actually in neurons, it goes out of the nucleus into the cytoplasm. And if we knock down Adnp, we lose neurites, we lose the axons, and we lose the dendrites. And here you see with Adnp in red, and then we lose. And if we add to cells that have lost their axons and dendrites because of oxidative stress or oxygen glucose deprivation, in red here it's lost, we add just NAP or CP201, the small fragment for Adnp, we repair. And how does it work? So NAP enters the cells by endocytosis. You can see it here in the movie, and you can see it here. It's a dynamine-dependent endocytosis, and dinosaur inhibits it. And NAP here is in red, you see. And the way it works inside the cell, so we have a lock and key mechanism, and it binds, it docks on the microtubule-end binding protein. And the microtubules are essential for the structure of the nerve cell and the structure of the axons and dendrites. And when they break down, as you saw, there are no processes and there are no cell connections. And the end binding proteins are essential for the microtubule dynamicity. And as you see here in the movie, you see that if we add NAP to a nerve cell, the microtubules move faster. You see it in red, and you can actually see it here tracked. And if we look at axonal transport in the live animal, and here it's by manganese-enhanced MRI, we see the movement. If you have ADNP deficiency, this is much slower. And if we add NAP back, we can protect. And this is what happens if there is a mutation in ADNP. So these are the most prevalent mutations in ADNP in the children. So these are shorter ADNP protein, it's heterozygous. These are the tracks of the microtubules. You see if you have full-length ADNP, all is well. If you have ADNP mutations, they are much shorter. If you add to these cells NAP, you repair and you have longer microtubules, longer tracks, and faster movement. And in terms of the dendrites and in terms of the dendritic spines, you see here that you have the dendritic spines are well organized in the ADNP normal animal. And if you have ADNP heterozygosity, so you have less ADNP, it's like a mutation, you lose dendritic spines, you give this animal for 9 days NAP, and you bring the dendritic spines back. And this is in the postsynaptic side of the synapse that we have billions of. And this is in the presynaptic side. Also, if we look at glutamatergic transmission, which is excitatory, so this is very important for our brain plasticity, you see this is the situation in the normal, and it's almost completely lost. I mean, it's really lost in the ADNP heterozygous mouse, and then we bring it back by NAP treatment. And this is in the hippocampus, and the hippocampus is much less developed in a child with ADNP syndrome as it should be in the normal situation. And the hippocampus, as we all know, is the area that undergoes degeneration, among other areas, but this is one of the first in the Alzheimer's brain, and this is the loss of the memory. And what we also know is that NAP enhances microtubule-End binding protein binding to the protein Tau, which makes the neurofibrillary tangles or the Tauopathy. As I said, that there are these two different pathologies in the Alzheimer's brain, the amyloid and the Tau. So what we showed is that by binding to microtubule-End binding protein and then to Tau, NAP or ADNP enhances Tau binding to the microtubules and protects against the formation of hyperphosphorylated Tau, which afterwards makes this neurofibrillary tangle. And what we found, so one of the major pathologies, as I said, in Alzheimer's disease is Tauopathy. And what we very recently found in a post-mortem brain of an ADNP child, we found Tauopathy, quite prevalent Tauopathy, as you see here, in comparison to the control. You see it here with the green arrows, and we can protect against this Tauopathy in tissue culture by adding a NAP and enhancing Tau binding to the microtubule, like what I showed in the previous slide. So if we take these haploinsufficient mice of us, heterozygous mice, that have half the content of ADNP, remember, if they don't have ADNP, they don't have a brain, and they are not born. But if they have only half the content, which mimics this situation of the mutations in the children, they don't learn. And they have to learn to find the hidden platform. We measure the time that it takes them to find the hidden platform. They don't learn. If we treat them with NAP, this is for 2 weeks, intranasally, they learn. And this is paralleled by increase in Tau pathology and then decrease when we give them NAP. And as you see, the mice also have Tauopathy, as we see in brain slices. And now, since we are talking about aging and the other side of the coin, so the other look at Alzheimer's disease, which I think also ties to the previous talk, we decided because there are de novo mutations during pregnancy or maybe in the germline, I was wondering if there are somatic mutations that occur in the aging and the Alzheimer's brain. And apparently I'm not the only one that wondered that. And what we discovered is that indeed, in postmortem Alzheimer's brain, there are mutations, somatic mutations. So they are not hereditary, but they occur probably with aging, and they occur in the brain. And ADNP has quite a number of mutations on it. We saw that by RNA sequencing, and we also validate it by digital droplet PCR. And what we saw is that the amount of mutation increases as the amount of Tauopathy increases. So this goes hand in hand with the mechanism that I've just shown you. And the other thing is that clinical trial was done with NAP in elderly people that suffer from amnestic mild cognitive impairment. And there it actually, and these people, it is known that they have tauopathy, and NAP actually increase their short-term memory, namely the ability to remember, this is shown here, the ability to remember an object that they have seen 12 seconds ago. So is Adnp the only protein that undergoes mutation in the Alzheimer's brain? No. We found mutations in thousands of genes. Remember, it's not hereditary, so it's not, the frequency is higher, but it's not like homozygous mutations. So, and we found it in all areas that we looked, in the olfactory bulb, in the hippocampus, in the dorsolateral prefrontal cortex, in the fusiform gyrus, etc. And we also looked, and this is data mining now, we looked also at single cell analysis, and we looked at endothelial, astrocytes, myeloid neurons, and you see that the most mutations were actually seen in endothelial cells, also in astrocytes and myeloid cells, sorry, and the least mutations were in neurons. So this is interesting. So this is actually also goes with the previous talk, saying that the support cells are very, very important. And here you see just the mutation number and mutation frequency increased in the Alzheimer's brains, and both in the cellular population and also when we look at the entire brain regions. And the mutation frequencies and number correlate again with tauopathy. And the tauopathy is actually called, this is BIRAX stage. BIRAX stage is actually the stage of how much tauopathy the brain has. And this has actually been correlated also with cognition. So the more mutation, the more tauopathy, the less cognition. And when we look specifically just in the olfactory bulb and to see what genes, can we group the genes together in families, we actually saw that many of the genes are associated with the cytoskeleton. Naturally, I looked for you because I really like tau and the microtubule. So the cytoskeleton, tau and microtubules is part of the cytoskeleton. So many cytoskeletal proteins are mutated. And then also proteins that are mutated in autism or in intellectual disability. So again, there is a correlation between these two ends of life, the beginning of life when something goes wrong, and also with the aging brain. And the cytoskeleton seems to play a key role, and it's very important in maintaining the structure and function of both the neurons and of course all the support cells. And when we look to see what proteins that are mutated, so genes that are mutated are shared, Adnp is shared between quite a number of brain regions. And here you see also in terms of the functionality, these proteins that are shared, but many proteins are different. So not the same protein will be mutated in one brain area or another brain area. And this is something I would like to go further into, and also to see how does that correlate with blood, because we're not going to kill the patient in order to diagnose him. We need to have something in the periphery in order to diagnose this process of mutation. So what I wanted to end is by saying that we are planning a clinical trial in the Adnp syndrome children, but that I think will open the way to other clinical trials that have a mutation in Adnp and have tauopathy, and see whether we can repair using our NAPVSIPQ, and we are well advanced into it. And these are our collaborations here. And I would like to thank all my students and again, the BIRAX for having this fantastic meeting. And thank you very much. And I hope I was clear. I tried to put everything in. Thank you.
Good, thank you. Thank you very much, Ilana. So we have time for a small number of questions. Can I start, Ilana? Do you want to take your screen down so that we can see other people?
Sorry.
Can I start asking questions? You had a very interesting experiment where you used the Morris water maze and exposed mice to NAP. I wondered, can you take those mice that have already developed tauopathy and then give them NAP, and does it reverse the tauopathy, or does just delivering NAP stop the tauopathy developing? In other words, is it a reversible thing? Is tauopathy a reversible thing, at least in the mice?
Well, so in this particular line, a mouse line, this Adnp haploinsufficient mice, we had to wait a very long time for the tauopathy to occur. So they were pretty aged. So it was actually age-associated tauopathy, unlike the human situation. However, we did see tau hyperphosphorylation early on, and the tau hyperphosphorylation is reversible. So what we know is in order for tau to deposit, it needs to be hyperphosphorylated, and we know we inhibit it. So I'm not so sure that once the tau deposits are formed, we will be able to dissipate it. But if we treat early enough, we will maybe avoid or prevent the tau forming the tangles. So I'm not sure that we can dissolve them, but we can probably, I mean, we know that we can inhibit them from forming.
Okay. So if anybody else wants to ask a question, please click on the raise hand button, and I can see you and invite you to ask your question. But so I'd like to ask another question. I know there's some, well, I don't know if it's a controversy, it's a question at least, about the relationship between cognitive function and tauopathy. Is there a causal relationship, or is it just a correlation? What's your view?
So what we know about Alzheimer's patients and the elderly population, we know that in terms of amyloid beta, you can have plaques and you can be as sharp as a knife. However, when you have tauopathy and when you go through the BIRAX stages, and the BIRAX stages actually say more and more and more, there is a correlation. Whether this is direct, whether the tau tangles will actually cause the disease, this is a question. However, what we do know is you lose synapses, you lose dendritic spines, you lose microtubules. This is actually Georges Perry's very old work. You lose microtubules in the brain, and you lose microtubules maybe because you lose some tau. And when you lose this, and you lose the synapses, this is a definite cause for cognitive impairment. So it is, maybe it's not direct, but it's indirect. The more tau you have, the more tau deposit you have, the more cell death you have, the more loss of synapses you have. So it is correlative, but it is also causal, because without synapses, we can't think. So this is...
Thank you very much. So I've taken up all the question time, and we need to move on to the next talk. But thank you so much for a really interesting talk. And it's so nice to hear about your proposed clinical studies. And I wish you well with those. Well, everybody does, I'm sure.
Thank you very much.
Okay. Thank you very much. Thank you.
Thank you.
So if I can move on to the third talk of this session from Adnp. So Anat did her PhD in the Hebrew University of Jerusalem in Israel, and is now at the Ben Gurion University of the Negev in Beersheba. And she's also trained in Northwestern University in the USA. And Anat is going to talk about neuronal regulation of proteostasis collapse in the worm, a model of aging. So over to you, Anat.
Thank you very much. I will just share my screen. Okay, good morning, everyone, and thank you, the committee, for inviting me to talk. I want to talk to you today about one of the hallmarks, of the 9 hallmarks that were identified as common denominator in aging and age-associated diseases as proteostasis collapse, and mostly focus on how this is regulated at the organismal level. So to start on equal level, I need to define proteostasis. And basically what we look at proteostasis as the sum of all the processes that are associated with the production of functional protein. So coming down from the ribosome, folding, degradation, and the end up of having a well-folded and functional proteins. All those processes are associated with a certain error rate, and this error causes the accumulation of misfolded proteins or metastable proteins, and that can contribute to a load on the quality control machinery. As long as this load is balanced by the machinery, the quality control machinery in the cell, we have protein homeostasis or proteostasis. Proteostasis can be disrupted under various stress conditions. I'm showing here the heat shock response as a sort of model organism for stress responses, and the principle is very general and shared by many stress responses, where you have introduction of damage to protein, and that particular damage is sensed by the quality control machinery. Quality control machinery being associated with the damaged proteins then activate a transcriptional response, normally because the transcriptional responses is somewhat associated, either binding or regulated by the quality control machinery itself. In heat shock response, the HSF1 is bound by chaperones. When they are occupied, it will trimerize, go to the nucleus, and activate transcriptional stress response that will lead to accumulation or to the production of more chaperone that can then rebalance the proteostasis environment of the cell. Problem is that we know many diseases that are basically showing dysfunction of this process, as we see accumulation of damaged protein in the cell. And one of the common denominators of these diseases is besides the association of damaged protein, is the connection to aging. Age of onset and age progression seems to be a major factor in those diseases. So it seems like the proteostasis network is able to cope with those proteins, mainly mutations that are there throughout the life when we are talking about mutation cases, but also sporadic. And then for whatever reason, we start seeing insufficient ability of the system to modulate protein folding later on in age. So one of the questions my lab is focusing on is what happens in aging and why do we see this proteostasis collapse? And you can imagine many scenarios that can explain this, and they don't have to be mutually exclusive. For example, one common accepted concept is the accumulation of damage over time, where cells become inefficient in clearing and damage is accumulating. We can also think of an example where the quality control machinery is becoming inefficient with age, and so more error are introduced and then more damage is introduced. And what I want to focus today is the notion that not only damage drive this process, but also regulation drive this process, and specifically that there are ways to regulate the quality control machinery, and that it's differentially regulated across time. And then if it's down-regulated, then maybe we can find ways to upregulate it or activate it again later on in age. So the question we started on almost 10 years ago is the proteostasis network differentially regulated with age? And a very simple experiment you will see through the talk uses worms' behavior in order to look at this process. And what we basically do is we grow worms until they reach adulthood. And then we start introducing them to stress. It's a population, each time a section of the population. So every time introduction to stress is naive animals that have not seen stress before. And we ask whether or not their stress response is similar across time. Originally, when we started these experiments, we expected to see a response very late in ages. Most of the damage we see is at later phases. But to our surprise, on the second day of adulthood, we saw huge decline in the ability to survive stress. So if we take a naive population and to worms, wild-type worms, and expose them to stress on the first day of adulthood, 80% survive. If we do this to their sisters on the second day of adulthood, basically we get 10-20% of the animal survives, and this remains. And this is not just survival, this is transcriptional response disactivation, because on the first day of adulthood, you can see here with a transcriptional reporter of the heat shock, we see a very strong activation of the heat shock response, and this becomes attenuated already at day 2 and 3 of adulthood. When we looked carefully at the transition phase here between day 1 and day 2 of adulthood, we could see that this is happening in a matter of hours, basically between 5 hours, between 7 to 12 hours after they reach adulthood. We see this decline, sharp decline in the ability to activate the stress response. if you look at the animal cycle of life and you ask what happened in this window, what you basically see is that the animals go through embryonic phases, for larva stages, become a fully functional adult. And then in this window, it's the activation of the reproductive system. So here we have a young adult that is yet to activate reproduction. And in the adult, we start seeing the onset of egg laying and production of progeny. This connection in timing, of course, was intriguing for us. And so we asked whether or not this timing is relevant and functional, and whether or not the reproductive system plays a role with this. And I need to introduce here two mutations. One of them is the GON2 mutation that basically impact the development of 4 cells that will give rise to the entire reproductive system of the worms. And then in the end, what you have is a sterile worm that doesn't have this somatic gonad. And this worm lives the same as wild type, but it is sterile. And then 2 mutations that were identified previously, GLIP1 and GLIP4, they affect the progenitor, the germline cell specifically. So you have a fully reproductive, developed reproductive system, but the germline are not proliferating. And so this worm is also sterile, but this lives longer. So we asked whether or not one of those mutations, and we of course took a panel of mutation looking at various processes in the reproductive system, have any relevance to this process. And we could show that while if you don't have the entire reproductive system, you don't see a rescue. If you impact proliferation of the cells, the germline cells, you see a complete rescue of this phenotype and basically maintenance of the quality control over time. This again was associated with the regulation of the heat shock response, where we could see now activation of the heat shock response at day 3 of adulthood and even later. And this is by reporter and also by quantitative PCR. You can see that the wild type loses its ability to activate various stress genes, whereas the GLIP1 animals that don't have proliferation of the germline do not. So to summarize this sort of introduction to the proteostasis collapse, what we basically see is that in a young adult animals, and it's young and it's just entering its reproductive phase, everything is fine, but then there is a regulatory mechanism that shuts off the robustness of the stress response pathways. And I don't have time to show you, but then also general quality control mechanisms such as muscle maintenance, various protein folding and metastable protein folding in the cell are also impacted. And we get fairly young adults, the first day of adulthood, but now the quality control system is much more restrained. And this is an epigenetic switch. Later on, Rick Morimoto and John Labadia lab showed very elegantly that in the young cells, we have the JMJD31 demethylase functional, and so the promoter is accessible and HSF1 can activate the stress response, and JMJD levels go down upon the onset of reproduction, and so the chromatin become locked, and the transcription factor may be activated but is not accessing the promoter, and therefore we don't have the activation of the stress response. So this explains at the somatic level why we don't see a stress response, and this demonstrates at the organismal level that the reproductive system is playing a role in this signaling pathway. But one of the questions we were left in and are highly intrigued with is how is this signaling mediated? So we have a signal coming from the reproductive system and going and in the end changing the fate or the behavior of the somatic cells, and we needed to understand how this signal is maintained. And we take various approaches to look at this, but one of them was sort of guided thinking into the idea that one of the major players or group of proteins that is majorly playing in this transition of onset of reproduction in many organisms is the gonadotropin hormones and receptors. And in insects, in other animals, there is a very close association of the gonadotropin family of receptors and reproduction regulation and metabolism. And we know that all of those are playing a role in this switch. So we therefore specifically ask if gonadotropin signaling pathway are involved in age-dependent decline and proteostasis. And so in C. elegans, if you look at the sequence, you can find 8 different members of this family. 3 of them have been deorphanized, and we know the ligand. And one of them has been studied in this concept and has both the receptor GNR1 and its ligands seems to be associated with reproduction. So we therefore decided to take this entire family and ask whether or not they have any impact on the quality control. And we started by our very simple experiments of looking at the second day of adulthood and asking whether or not we have activation of survival or survival in various mutation. We found 2 candidates that were showing rescue behavior on the second day of adulthood, and their quality control was not affected on the first day of adulthood from this family. And we decided for various reasons to start focusing on GNR2. So the first question we ask is whether or not GNR2 is actually expressing the reproductive system, or is it neuronal? And we can show that it's expressing specific neuronal cells in the body. There are several cells in the head, several cells around the vulva that I will discuss in a second, and then some in the tail. We then also looked at total reproduction capability that was not affected, and lifespan that was also not affected. If you look at the cells that affect the reproductive, they sit on the vulva and they basically regulate egg laying. So we tested whether egg laying is modulated, and we can show that indeed the rate of egg laying was reduced when we have a GNR2 mutation. So of course the question then goes, what happens to survival over time, and whether or not it's really specific by GNR2. So to test that, we used another mutation in GNR2, and so the same behavior, and then used RNAi to knock down the GNR2 gene, and again, so the same thing over time, rescue of the proteostasis. We've looked at other various responses and so modulation there, but again, I will focus on HSF1. Here we can quantify the activation of the Hitchcock response at day 1 and day 3 of adulthood, and whereas both are activating the Hitchcock response at day 1 of adulthood, at day 3 the wild type animals no longer do that, but GNR2 mutant activate the Hitchcock response and activate various Hitchcock genes as exemplified here. I call it quantitative PCR. We next ask if we can identify the ligand, and when we looked at the GNR2 and did 3 comparisons of sequences, we could see that it belongs to the corrosinine family inside the gonadotropin family. And so we looked at the consensus sequence for corrosinine, and then used this to search for the proteome of the worm, and we could identify several candidates, but the top one was NLP23. It has 3 different peptides. This one was not shown in animals, but it is very similar in conserve and maintain many of the conserved sequence. And so we looked at that as a possible candidate putative ligand for GNR2. So the first thing we ask is whether or not if we delete NLP23, we see the same response. And the answer is yes. If you delete NLP, either by RNAi or by mutation, you again see rescue of the proteostasis. And if we have a double mutant of NLP and GNR2, we don't get more rescue of the proteostasis. We get very similar, suggesting that they're functioning in the same pathway. We then again look at the Hitchcock response activation specifically, as then you can see that 2 different GNR mutation and also NLP activate the Hitchcock response on the 3rd day of adulthood, whereas the wild type does not. Finally, because we had the egg laying phenotype, we asked whether the egg laying rate is also affected, and we could see that it is, although not to the same extent, and that the double mutant again is similar, not significantly different than the single mutant. So then we ask specifically on the regulation, and this is basically where I want to end, is whether or not those two are regulating in a similar manner. We have done a lot of work here. I'm just summarizing a few examples. So first, there are both the GNR2 and NLP are requiring DAF16. Both of them require GMJD, so this puts them in the gonadal longevity pathway, this and more data. But then they are not entirely the same as if you look at PQM transcription factor, one requires it, the GNR2, and NLP23 not. So this could suggest that either it's partially activating, or there are other factors, or it's not completely the ligand. You can see here that the DAF16 is entering the nucleus in both GNR2 and NLP, but PQM only does that in GNR2. So to summarize what I told you is we found two gonadotropin-like hormone receptor, GNR2 and 6, that modulate proteostasis collapse in adulthood. GNR2 also modulate reproduction, which tie this to those processes. GNR2 act downstream of the gonadal longevity pathway. I showed you some of the data here, and that both are expressed in neurons. There is another paper on GNR6 again showing this very nicely. NLP and GNR2 acts in the same genetic pathway, but we do not know if NLP is the true ligand of GNR2, or whether or not it's a possible ligand of other GNR6 or other receptors. And with that, I need to thank you for listening, and thank, of course, the students that did the work. Most of the work you saw was my student, Maud Kirchner. She was helped by various members in the lab, and this project started with the work of Libat Rebaz. And of course, I have to thank the foundation, the ISF, Ministry of Science for Fellowship for More, and then my collaborator in this BSF grant, Ehud Cohen and Vina Prahalad. And thank again you.
Thank you very much on that. So you could take your presentation down. You can see the participants then. Good, thank you. So Ilana has a question. Over to you, Ilana.
So I remember the days when Stephen Kufler actually found the GnRH is a neurotransmitter many years ago. So, and this is GnRH receptor that you're talking about. I hope I'm not mistaken. So the question is how what you're saying translates to the human or the higher organisms. And also taking into consideration, you know, that these proteins, I mean, are very important in the reproductive idea, and also in maybe in the function of the brain with the neurotransmitter function.
So working in C. elegans, we always get the very important question of how our work is related to. So to summarize it in a few points, proteostasis collapse or proteostasis decline is shared by many model organisms. As I said, it's a hallmark of aging. Proteostasis collapse was recently shown by very elegant work by Reut Shilgi's lab in the Technion to also be in senescence human cells. So you can see that the heat shock response is not activated in senescent cells as well as it was in young animals. So this suggests that it occurs. She shows it on the cellular level, which then, that's the fact that the cell can or does that, and the regulating mechanism basically determine when. Okay, so that's on the
GnRH. This is what interests me. I agree with you.
On that we don't know. The gonadotropin related or associated or family is very big. Their role in insect is not exactly the same as human. As I said, some of them are regulating metabolism and some aspect of reproduction. This is not the function that you will see as a pituitary gland and so on. But this could be the local regulation that you do see in some of the aspect of the levels of the reproductive cells themselves. They're amazing family. I mean, they can regulate in ants who is a worker and who is a queen, and then the change between worker and queen when that is required. So they're really determining the onset of reproduction that ties this very nicely to what we're talking about. Whether or not the onset of reproduction is also the main driver in human aging, I'm not going to go there. I think it's very early, but I think that we're talking on a very basic level of highly conserved processes, metabolism, reproduction, and quality control that has to be tied together, and so they will play a major role in the process of aging.
Okay, thank you. Thank you so much. I think we've run out of time now, so we need to move on to the break. I think we have just 10 minutes left for break, and the second session starts at 1/2 the hour, whichever hour you're living in. So thank you very much to all the speakers this morning. They were fantastic talks, a great introduction to this meeting, and I'll hand over now.
Thank you, Karen, so much for chairing the session. We actually have a 15-minute break today. Tomorrow we'll have a 10-minute break. So I'll release all of you. I do want to tell you that I'll be showing, I think, a really fabulous clip of Israel. And so this will whet your appetite for those of you who couldn't arrive to Israel these 2 days, so you'll be able to see it during the break. I also, before I let you go, I do want to acknowledge Dov Shmotkin, who just handed over the reins to Sylvia Cotton. And I see both of you here, so I want to give a shout out for running the Herzig Institute of Aging at Tel Aviv University. So thank you, Dov, to you for all you've done, and for Sylvia, for what you're going to do. Okay, so enjoy the break, and we'll see you in 15 minutes. הצלחתי להתחבר. 2 more minutes and then we'll start. I'll introduce our next chair and then our chair will take over. I hope some of you got a chance to see the clip from Israel, just to whet your appetite. Good wine, good restaurants, none of which are open right now, but they will be. Okay, everyone, welcome back. I hope you had a nice little break. You didn't have to go too far. So it's a real pleasure for me to introduce our next chair, who is Dr. Richard Siow, and I'm also from KCL. And as well as working on vascular biology, he's also director of the aging research at King, called ARC. And he's interested not only in the major research that he does, but also in the social impact of aging. So that's quite a variety. So thank you, Richard, for joining. And let me just remind all of you that the rules are the same. If you have a question, go into the participants box and raise your hand and the chair will call on you. Everybody is on mute. So when it comes time for you to ask your question, please unmute yourself. Thank you. Thank you, Richard.
Karen, thank you very much. And also colleagues for organizing this wonderful 2020 virtual BIRAX conference. I'm really glad to see many friends and colleagues in the audience who were also in London at the 2018 BIRAX conference, where we launched the aging theme at King's College London, which you can probably see just over my shoulder. It's a view of London from my helicopter where I am at the moment. So it's great to be back in Israel virtually and to see you all at this conference. So our next session will be, again, a mix of Israeli and British speakers, starting off with Ehud Cohen. I met Ehud maybe 3 or 4 years ago in London for the first time. Ehud is faculty member of the Institute of Medical Research, Israel, Canada, at the Hebrew University of Jerusalem. He holds the Henry and Erna Lehr Professor Chair for Research in Neuroprotective Diseases and is the director of the Brain Disease Research Center of the Faculty of Medicine at the Hebrew University. So I'll hand over to Ehud, who will be speaking about his research on brain aging. What goes wrong with aging? Thank you, Ehud.
Thank you very much. Thanks, Karen. Thanks, Richard. I'll just share my screen with you. I hope that you can see it all. Okay, and I also want to thank my colleague, collaborator, and friend NAPVSIPO for providing great introduction, because I'm going to take it pretty much from where Anat left us with, talking about the proteostasis network. And I decided to use my 15 minutes to focus on one ongoing project, which is not published, but I'll start by giving you a short introduction. So,
Ehud,
yeah.
If I can introduce, there's a little bit of an echo when you speak. I don't know if it's the earphones or something, but it's not as clear as it could be.
Is it better?
About the same.
And I'll try to speak less loudly. Maybe.
Okay, thank you.
Okay, I know that Anat, or I guess that Anat is going to use this slide, which was published by Soto in his review in 2003. I just put it back in order to focus on two specific brain slices, which were taken post-mortem from patients that suffered from two different disorders. Alzheimer's disease, as was mentioned by Rana as well. These individuals have the A-beta plaques as well as tau tangles in the brain, which were mainly composed of the aggregated peptide A-beta, and of Huntington disease, which actually represent a group of at least 9 polyQ expansion diseases, which certain proteins harbor abnormally long polyQ or polyglutamine expression. I'm going to go back and forth between these two models. However, the accumulation of toxic protein aggregates is not the only common feature of neurodegenerative disorders. As you all know, they also manifest late in life. And just to give you an example, this is a table which was taken from a paper which was published back in 2015 by Bartis Trooper Group, and it shows that individuals who carry Alzheimer causing mutations, they develop the disease in the 5th or 6th decade of life. This raised the question of what goes wrong with aging and why Alzheimer's disease onsets late in life, just like other neurodegenerative disorders. And more importantly, is it possible to delay the onset of Alzheimer's and other diseases by slowing the progression of aging? How can we slow the progression of aging? So there are several.
Ehud, I'm sorry to cut in again, but we really can't hear you well. So maybe talk without the earphones.
I will try.
Okay, because yesterday when we practiced, I heard you loud and clear. Sorry about that. I don't like to interrupt, but I prefer everybody gets your message.
Is it better now?
It's a little better.
Okay. So we heard before about caloric restriction or actually dietary restriction, but probably the most prominent aging regulating pathway is the insulin IGF signaling cascade. And just to give you a glimpse, Cynthia Kenyon's lab published in 1993 that C. elegans that carry mutation in DAF2, which is the sole insulin IGF receptor of the worm, lives as twice as long as the wild type animal. This was shown to be conserved in mammals as deletion of one copy of the IGF-1 receptor extended lifespan of mice, and most likely also in humans because centenarians were found to have functionally significant insulin-like growth factor as published by the group of nearby lab. How does it work? So in the worm, we have the DAF2 receptor, which, as I mentioned, the sole insulin IGF-1 receptor. It negatively regulates a series of transcription factors, including SKIN1 NRF, OXO-DAF16, the heat shock factor 1. So if you knock down the activity of DAF2, you hyperactivate the transcription factor. They're now free to enter the nucleus and regulate the target genes and to slow the progression of aging. And functionally, if you look at lifespan, you can see that DAF2 mutants that actually exhibit very low activity of DAF2, they live more than twice, actually 3 times longer than their counterparts that have the wild type copy of DAF2. So now we have a tool to slow down the progression of aging, and it opens the way to ask the question of whether the inhibition of IGF signaling, thereby slowing the aging, can protect from proteotoxicity. And I'm going to introduce you with the first model that I'm going to use. These are C. elegans worms that were created back in 1995 by Christopher Link's lab. And what they did, they cloned the human A-beta, one of the aggregated peptides which is associated with Alzheimer's, one of the A-beta family, actually, under the UNT54 promoter. This promoter drives the expression of A-beta in the body wall muscles of the worms. And as a result, it won't become paralyzed. It can only move its head, as you can see right here in this microscopy image. This worm cannot move, so it consumes the food within the reach of its head. This is a phenotype that can be followed, and it's indicative of proteotoxicity, of the toxicity of aggregated A-beta. So if we knock down DAF2 to hyperactivate its downstream transcription factor, you can see that the rate of paralysis within the worm population is significantly and dramatically lower compared to control animals. So if we look at day 12 of adulthood, nearly 60% of the animals that were not treated show the paralysis phenotype, while only about 15% of the DAF2 RNAi treated. The question is, how does it work? And it brings me to the description of proteostasis network that Anat mentioned briefly. As you all know, right after translation, the polypeptide that is newly synthesized has to undergo a series of events in order to fold properly and become functional. After the life cycle of the protein, or when it's unwanted or damaged, it has to be recycled by either the proteasome or autophagy. However, not all polypeptides reach the mature functional folding. Some of them misfold, and there is a whole series of mechanisms that maintain proteostasis by sorting those misfolded polypeptides, sequester them, and eventually degrade them. And many of these steps are negatively regulated by aging, as shown by a series of plethora of studies. So the proteostasis network supervises the integrity of the proteome. And the question is it possible to preserve proteostasis through late stages of life and then preventing the accumulation of aggregative protein and prevent or at least delay the onset of neurodegeneration? And here I want to focus on two questions. So it was shown by us and by many other labs that knocking down the activity of aging promoting pathways or inhibiting aging by dietary restriction preserves proteostasis and protects the animals. And when I say animals, it was shown also in mice that from neurodegeneration. But the question that I want to raise here is, does the proteostasis network equally respond to this similar proteotoxic challenges? Or maybe perhaps the proteostasis network tailors its response according to the proteotoxic challenge that cells encounter. And if there is such tailoring, how this response is coordinated at the organismal level. What was the rationale that was underlying this project? We asked, is it possible to identify a single chaperone that plays opposing roles in the face of different proteotoxic proteins? Meaning that if we knock it down in a model, C. elegans model that express a certain neurodegeneration causing aggregative protein, it's protective, and in the face of another challenge, it's actually deleterious. So what were the second model that we used? The second model was actually one strain, which was created by the lab of Rick Morimoto back in 2002. And those animals express polyQ attached to YFP, meaning that they have an aggregative protein, which in humans associated with, as I mentioned, at least 9 neurodegenerative disorders, including Huntington, and they can be visualized by a fluorescent microscopy. When they tested how the aggregation and toxicity of polyQ is affected by aging, they found that if you look here at Q35, as the animal aged, the number of aggregates increased and the motility actually declines in an aging-dependent manner. So what is the proteotoxic phenotype that can be followed? In order to measure the rate of proteotoxicity, which is promoted by the expression of polyQ, we use what's called the crushing assay. And the idea is that you soak the animals in a drop of liquid and they start kicking. So you can count as kicking per time and see whether the number of kicks increase or decrease, as this is indicative of proteostasis. So in order to compare and look for chaperones that play opposing roles, we used 4 worm strains. 2 of them express the polyQ35 under the UNT54 promoter that drives the expression of body wall muscles, and 2 and the other one, sorry, the Q35 is expressed under the regulation of the RGF promoter that drives the expression in neurons. The very same promoters we use in order to express A beta either in neurons or in body wall muscle. Then we ask whether we can find such chaperone that it's knockdown protective in one worm strain and deleterious in the other one. And to cut a long story short, this work was done by Hannah Buchholz and Philippa Rahal-Marx in my lab, and they found that a protein, a chaperone, which is called torsin A, if you knock it down, it protects the animal from the paralysis phenotype, as you can see here in black. Here is the paralysis rate of wild-type animals untreated, and here of animals that were treated with torsin A RNAi. What is torsin A? It's a member of the AAA family of proteins that includes some Hitchcock proteins. In humans, mutation in torsin A leads to the development of dystonia as an inherited muscle disorder. It was shown by the group of Guy Caldwell that the overexpression of TOR2 protects the worms from poly-Q and alpha-synuclein, as you can see here in the title of this paper. So first, we wanted to see whether the knockdown of TOR2 and TOR1, which are the two ortholog of torsin A and C. elegans, equally affect the proteotoxicity in neurons and muscles. You can see in here that when we knock down either TOR1 or TOR2, specifically by RNAi towards the treatment UTR, we got similar effects in neurons, and the same phenotypes were seen when we used mutants that have deletions in TOR1 or 2, meaning that knockdown of both proteins, TOR1 and TOR2, is deleterious when the animal expresses poly-Q. On the other hand, when we looked at the rate of paralysis, and in this case, this is in neurons, we saw that the knockdown of torsin A or of each of the torsins protect the animal from the paralysis. So knocking down torsins is deleterious when the animal expresses poly-Q and protective when the animal expresses A beta. Then we ask, what is the mechanism and how it works? And in order to address this, we ask which genes actually respond in opposition to the knockdown of torsins in worms that express A beta, the CR2006, or worms that express the poly-Q35. And what I'm showing you here is actually comparison of the response difference. We look for genes that the expression is going either up when poly-Q is expressed and down when A beta is expressed, or vice versa, when poly-Q is downregulated here and A beta is upregulated. Sorry, and what you can see here that we got several lists of list of genes that include about 30 genes each, and I'm going to focus on the most significantly responding or opposingly responding genes, and those are actually 3 neuropeptides and 1 additional gene. And when we looked at the biological processes, we found that neuropeptide signaling, cell adhesion, and messenger RNA splicing are the 3 processes that respond in opposition. So how the knockdown of these neuropeptides affect the physiology of the worms? And again, when we looked here at A beta worms, knocking down each of those 4 genes, the 3 neuropeptides and WO2, you can see that there is reduction in the rate of paralysis, meaning that the animals are relatively protected. However, when we looked at worms that express poly-Q, we got lower number of body bands, meaning that proteostasis is enhanced. And it was very similar when we knocked down 2 of the neuropeptides together by multi-silencing cassette, or all of them together, meaning that each one of them is equally important. Which neurons express those neuropeptides? So again, to cut a long story short, we characterized all 4 genes. I want to show you that what we did is actually creating worms that express PD2-mater, the red fluorescent protein, under the regulation of, in this case, WO2, one of these 4 genes. We cross them with animals that express GFP in no neurons, and we define sets of neurons. Many of them are known to be serotonergic neurons or neurons which are involved in the regulation of the Hitchcock. Is it surprising? Actually, it's not, because back in 2011, it was shown by Morimoto laboratory that neuronal circuitry regulates the response of C. elegans to misfolded proteins. And later on in 2013, we have shown that the GPCR receptor, which is expressed in neurons, actually also regulate proteostasis in A beta mode. So is the insulin IGF signaling cascade involved in here? I just want to show you one piece or one set of experiments in which we knock down DAF2 and we ask whether the expression of torsins is affected. And the answer is yes. When you knock down DAF2, torsin expression is elevated by about 60 to 70%. And when we mixed RNAi towards DAF2 and one of the transcription factors, DAF16, ADCF1, or SKIN1, we found that DAF16 and SKIN1 are critically important in order to mediate the elevation in expression level upon the knockdown of DAF2, but not ADCF1. And recently, just last week, we found that 2 of the neuropeptides, that their expression is regulated by DAF2, while 2 others are not. This means actually that DAF2 and this aging regulating pathway plays an important role in coordinating this proteostasis maintaining signaling mechanism in the womb, but it's most likely not the only one. So just to sum up, there are several groups of neurons that when somatic cells sense proteotoxicity, they send a signal, which we don't know exactly what it is. That signal is received by sets of neurons, which integrate it in order to release the response, which is specifically tailored according to the type of proteotoxic challenge. Those neuropeptides in this case activate gene expression, including of IIS or insulin IGF signaling targets. I can show you the data, but this is what we find. And as a result, we get proteostasis enhancement. There are many open questions that we have to address. First is, how does the cell, the somatic cell, inform neurons what the challenge it faces, and what are those signals? What are the other messengers which can be sent or released by neurons in order to activate a different program of proteostasis maintaining gene expression? And how the signal modulates gene expression in the target cell? And I'm mainly focusing or referring to genes which are not IIS targets. The importance of this project, in my mind, is that it shows that we cannot actually talk about activating the proteostasis network in response to neurodegeneration, but we have to categorize different disorders, understand what are the right signals that activate the specifically tailored proteostasis modulating program, and then develop treatments which are specific to subtypes of cases in one disease, or and obviously different disorders. I'll finish with that. I'll just thank people of my lab, former members, the funding agencies and collaborators, including NAPVSIPO and Regulator, and thank you for your attention.
Thank you, Ehud. Maybe if you could stop sharing your screen, we can see each other.
Of course.
Because of time, we can only take 1 or 2 questions. I see that Ilana Gozes has a question. You've got a hand up. For the others, if you'd like to ask a question, please use the participants button and then raise hand function.
Oh, Ehud, can I speak, please?
Yes, go ahead.
Yeah, sure.
So Ehud, thank you for a very good talk, as usual. And I have the same question that I had to Anat, because I'm interested more in the mammalian brain or the human brain. And we know, so you spoke about neuropeptides, and we know that somatostatin, that's known from many years ago, goes down in the Alzheimer's brain. And other peptides like VIP, my favorite, also goes down. So the question is, do the peptides, the neuropeptides that you are, or the, yeah, you said neuropeptides, that you are talking about have homologs in the mammalian brain?
So I will start by referring to the question that you asked, Anat, about neurotransmitters, it was shown by Patricia Messiel from the University of Braga in Portugal that serotonin, for instance, is critically important to protect mice that express polyQ in their brains. So at least at the level of neurotransmitters, we know that there is a conservation in mammalian brain. I don't want to say humans because the evidence there are not that strong, but in the mouse, it was very elegantly shown. Now, regarding neuropeptides, there are, the similarity between C. elegans and humans is kind of weak because these are two creatures which are pretty far away in evolution, as we all know. However, many basic mechanisms have been shown to be conserved. And this includes, of course, the RNAi or microRNA in humans and apoptosis and many others. There are no obvious orthologs in the sequence level to those neuropeptides, but I wouldn't be surprised if there are functional orthologs, and I have to say that these are the very early days of the exploration of inter-tissue communication, and most of the information that we have in the moment is from simple organisms, mainly C. elegans. So it's very likely that there is, but I think it's too early to make strong statements here.
Thank you.
Ehud, thank you very much. I think there are no further questions for you, so it leaves me to thank you again for a great presentation. And because of time, we'll move on to the next speaker. Good to see you again. Our next speaker is on a slightly different subject area, is from Sharon Toker, who's head of healthcare management program at Tel Aviv University Koller School of Management. She's associate editor of the Academy of Management Perspectives and Journal of Occupational Health Science. She was awarded an Early Career Award by the American Psychological Association and National Institute of Occupational Safety and Health. So I'll hand over to Sharon for her talk on the depressive price of being a sandwich generation caregiver. So Sharon. Is Sharon there? I probably need to switch on your camera. If you're there, I don't see Sharon. Karen, do you know if Sharon is with us?
I actually don't see her either. So
I think I saw her earlier, but maybe she's dropped out.
Interesting. Okay, so I think I don't see her.
Yeah, maybe she'll reconnect.
Okay, so
maybe we can move on to Dinah.
Our next, yeah.
Dinah Zur.
And
that's okay. You are online. I can see you. So let me quickly introduce Dinah. Dinah is a retina specialist and from the Department of Ophthalmology at Tel Aviv Sharansky Medical Center and a faculty member of the Sackler Faculty of Medicine at Tel Aviv University. Her research is on retinal and choroidal imaging, retinal vascular diseases, and hereditary retinal diseases. And she's one of the initiators of the International Retina Group and coordinator of continuing medical education in ophthalmology at Tel Aviv. And Dinah will be speaking about algorithms for diagnosis and management of age-related macular degeneration. Thank you. I'll hand over to Dinah. And thanks for stepping in.
So thank you very much for the invitation and letting us present our project here today. But before talking about our project, let me take you back to anatomy class for a moment. The eye is a complex organ which actually focuses light through the cornea and the lens onto the retina, which is the inner lining, which is light sensitive in the back of the eye. This is what the retina looks like when we look into the eye. And it actually works like a digital camera. It transforms optical information into electronic information, which goes to the optic nerve and then to the the visual cortex, which is the area in the brain that is responsible for vision. In the center of the retina, there's the area which is called the macula, and this is responsible for the central vision. So if you have a problem in your macula, this is how you will see the Mona Lisa. Today, we have a very efficient imaging modality, which is called optical coherence tomography, OCT, which allows us to take, within seconds, high-resolution images of the macula that reach almost histological resolution. Now, let me explain a little bit about the disease, age-related macular degeneration, or AMD. It is responsible for the majority of blindness in the Western world in the elderly population. Has a global incidence of 10%, and the incidence is growing with age. And this is not getting any better. Today, we face 196 million people with AMD in the world. And in 20 years, this number is expected to grow to 288 million. And this is what it feels like to be an AMD patient. Those patients suffer not only from this decreased visual acuity, but also very bothersome distortions of the vision. It impacts the central vision, as you can see here, which is responsible for reading or the ability to recognize faces. And as you can imagine, it impacts many daily activities of life. It impacts the independence of our patients and actually quality of life. Now, AMD can be divided into two forms: the earlier non-neovascular dry AMD, which is responsible for 90% of the patients. And we can find some signs of dry AMD in almost 1 of 3 elderly patients. But this form is asymptomatic in most of the cases and can be slowly progressive. In contrary, neovascular or wet AMD is responsible only for 10% of the cases, but it is responsible for almost 90% of blindness caused by AMD. And it causes an abrupt vision decrease in vision, which needs to be urgently treated by injections. The hallmark of the dry AMD form are the drusen, which are yellowish deposits in the macula, as you can see it here. And using OCT, we can easily image those. And you see this waviness below the retina, which represents those drusen. The neovascular or wet form of the disease, in contrary, is characterized by proliferation of neovascular vessels beneath the retina. Those vessels are very permeable and they leak and cause hemorrhage in the macula. And contrary to the dry form, this causes an acute decrease in visual acuity. And using OCT, we can display the disease with fluid in different retinal compartments. So the management of the disease is very time intensive. Patients need to be seen monthly using this OCT imaging, ophthalmology exams, and injections need to be given in a high frequency, monthly, sometimes over years or even lifelong. But let me explain to you a little bit better what this means for our patients. They actually have to come into the clinic at least once a month, most of the time. And this is how a calendar of my AMD patients look. And each of these visits may last hours of waiting and therapies and imaging at each visit. But there are many more limitations and burdens for the patients and for us. As it is a chronic disease, adherence to treatment might get low. Patients are elderly and visually impaired, and that's why they frequently depend on help, mostly from younger family members that need to bring them in at the clinic. They miss injections not only because of this, but also because they have other health issues than AMD. They have a lot of comorbidities with age. There's a lot of anxiety related to the injection itself, but also because of fear of losing vision. And practical issues like lack of transportation, of lack of accessibility, which is even more pronounced today during this pandemic. But also money plays a role. These treatments are not all reimbursed, and patient affordability plays a role. And of course, all of this causes a high burden, not only on the patients, but also on our health systems. So if we would have talked about the disease 15 years ago, the talk would have been another, because back then there were no efficient treatments available. Today, with the injections that inhibit VEGF, vascular endothelial growth factor, the picture has changed, and we actually can maintain visual function for almost all patients. in the first 1 to 2 years with frequent injections. However, when we look at the long-term prognosis of the disease, it looks much less promising. As you can see, over time, at the beginning, there's initial improvement in vision, then we reach some plateau. But even with the best standard of care, patients lose vision over time. And if you compare the baseline vision with the final outcome after 5 years, in patients that were in randomized controlled trials receiving the best possible care, still they finish with worse vision than they started with. Another problem is the high burden that this disease puts on us physicians. At each visit, we actually have to go through multiple scans of one patient, analyze them, quantify them, and then compare them to the previous visits in order to decide if this patient has an active disease. Why is this so important? Because actually the whole treatment of the disease is guided by the presence or the absence of fluid, as you can be seen on OCT. So each time the patient comes in, as you can see here at your left, I have to go over the scans and decide if this is an active disease, if the patient needs treatment, at which frequency he needs this treatment, and if we have to change the drug, and so on and so on. And this management has lots of limitations, which has been shown previously. We assess the fluid currently in a manual qualitative manner. This is, of course, operator dependent. And if another retina specialist will go over the scans, he might disagree with me. And also, we as humans are not capable to quantify this fluid. Because of a lot of change in the last 100 years in ophthalmology. And these days, we're able to use the potential of artificial intelligence in the management of the disease. Today, we have automated algorithms that allow us to detect the fluid on the OCT and also to volumize it per retinal compartment. So let me show you again. In the upper part, you can see a regular OCT scan as I look at it today in the clinic. But using an automated algorithm as the one that was developed by Notal, an Israeli company, which is called the Notal OCT Analyzer, or NOAA, we actually today are able to use the potential of machine learning with this fully automated algorithm that provides us with anatomical analysis according to the retinal compartment and gives us a quantitative output per patient at the special time, but also over time in comparison to previous visits, and gives us a much better and sophisticated way to look at these patients. I will try to explain to you how this actually works. We have the OCT file, as I showed you before, which is actually a volume scan. There are lots of scans at every visit. The algorithm does an image processing, and then using artificial intelligence and machine learning, we get a classification and a quantification of the pathology in each different compartment, as you can see here in different colors in this little video. And in the end of the process, we as doctors can have an individual patient report, which gives us not only information about this certain time of the patient, but also in a graphical representation with longitudinal reports over time. Another advantage of the algorithm is that we can now do a big data population analysis. So if you have one patient, you can plot him on this graph, as you see here below in the picture, and then with a high probability know which disease he is suffering from in comparison to the population in the database. Previous collaboration between Tel Aviv Medical Center and Belfast Queen's University has shown that this algorithm reaches a high accuracy, sensitivity, and specificity when comparing it to the gold standard, which is today the evaluation by retina specialists, which was over 90%, and this was published in Ophthalmology in 2016. So our project aims to examine the long-term functional and morphological outcomes in AMD patients who are treated with anti-VEGF injections using this NOAA algorithm. We aim to investigate the performance in a clinical setting of the NOAA algorithm to monitor the disease and also detect progression of the disease. In the end of this process, we are aiming to build models that incorporate demographic factors, all the available data on comorbidities, treatment rates and types and drug types in determining the functional outcomes. Let me introduce you to our team. This is a collaboration between Queen's University and Belfast, Tel Aviv Medical Center, which is affiliated to the Tel Aviv University, and Notal Vision. The principal investigator at Queen's University is Professor Tunde Peto, who is a very well-experienced retina researcher and a specialist, and she has been working on the previous work by validating this algorithm for AMD. In Tel Aviv, Professor Anat Lowenstein is one of the well-known retina specialists in the world. And she was actually one of the pioneers who was involved in the development of this algorithm long before the ophthalmology community was even aware about the potential of artificial intelligence. And all of this would not have been possible without Notal Vision, with Gidi Binyamini and Moshe Chavilio, who were the pioneers and developed this fully automated algorithm as the first to detect fluid on OCT. Now we see a large potential of this collaboration due to the large clinical and imaging database, which we will set up from these two centers. We will have more than 8,000 patients, which means more than 16,000 eyes, with a very well documented long-term follow-up of more than 5 years, which means more than 150,000 visits, which means more than 9 million scans for evaluation. This is the workflow of the project. Both of our centers are first identifying all the relevant patients with neovascular AMD who are treated and follow up in our clinics. We are then extracting the long-term clinical data. Also, we are, of course, extracting all the OCT images, as I showed you before, which will be evaluated by the algorithm from Notal Vision. And in the end, we will have this huge data set, which will allow us to do an analysis of combination of the functional and anatomical data with the treatment types and frequencies. So we see the potential here because it will allow us to better understand the morphological, functional, and demographical parameters in order to provide a practical base for better disease management. We will also be able to visualize the disease morphology, which will allow us to earlier detect the disease and allow for better and efficient therapeutic follow-up. We also think that using this algorithm, we will be able to reduce the burden of care for the patients and for us, and also improve real-world clinical performance, which I showed you before, is still disappointing in the long term. Also, it will improve physicians' decision-making and rethink the current treatment algorithm, which will now be based on quantitative measures. And in the end of the day, we hope that we will be able to individualize and customize the treatment for the individual patient and improve outcomes. So on behalf of all collaborators, I want to thank you very much for your attention. I will be glad to take your questions.
Great, thank you very much, Dinah. Any questions for Dinah? Please raise your hand in the participants box using the raise hand function. Maybe I can start. This area is very interesting to me as a vascular biologist interested in the microcirculation aging. Have you correlated OCT with dynamic vessel analysis, looking at the functional properties of the retinal microcirculation?
Actually, we do have now the ability to visualize the flow in vivo using an OCT angiography, which is a relatively new tool, and it does not need the injection of dye as we were used to do it previously. This technology is relatively new and quite limited for some questions. And in this certain project, we will not assess the flow in the retina, but I'm sure that Notal will go a step further and we will incorporate this in the future.
Okay, that's great. Thank you very much. I also know the Belfast team at Queen's. We've been discussing some of this. Any questions? No one's raised their hand so far, so maybe I can ask one more. What is the value of OCT and, in general, retinal microcirculatory function for prediction of, neurodegenerative diseases, not only eye diseases and retinal diseases?
So that's an important question because we now know that using the OCT, we can detect a lot of more than eye diseases. There's a big work done on Alzheimer, for example, or Parkinson, which was previously published, where you can, using, again, artificial intelligence and big data, identify biomarkers for Alzheimer on the OCT of the macula and the optic nerve.
Great, thank you. Very interesting. I think this is very important for looking at biomarkers in the community using non-invasive techniques. Any questions from the audience? If not, because of time, we can move on. I'm glad to say that Sharon Toker has joined the meeting. As I've already introduced Sharon, although she wasn't here. Maybe I won't go through that again, but maybe just to mention that Sharon is the head of healthcare management at Tel Aviv University College School of Management. Sharon, if you could share your slides, please. You've unmuted yourself. And again, the title of her talk is The Depressive Price of Being a Sandwich Generation Caregiver. Thank you, Sharon.
So thank you, and please accept my apologies. We had an accident this morning. My daughter had an accident, so I was offline, but now I'm here. So today, I think it's a bit even related to the theme of our talk. I'm going to talk about the depressive price of being a sandwich generation caregiver. And we'll ask ourselves, what is the role of organizations and managers in helping those that care for those in need? So let's start with the definition. Sandwich generation caregivers are those employees who take care of both their children and elderly family members. These actually can be elder or their spouse or a sick sibling, so they do not necessarily have to be aging persons, but usually they are. And they have to simultaneously strive to accomplish their work goals. If we talk about the prevalence, we see that the percentage of employees which have to take care of both children and parents is quite high. And this number is expected to grow in the near future due to the growth in life expectancy, the aging of baby boomers. People are postponing the beginning of parenthood. So we know that there is going to be an increase in these numbers. But let's add some complications to this issue, and we'll talk about the coronavirus. So we know that sandwich generation caregivers during the COVID-19 pandemic have to take care, first of all, of themselves because they're afraid to catch it. But if we think about their work settings, many of them work from home. They deal with problems that relate to transportation, social distancing at work, downsizing, layoffs. On top of that, they have to attend their children, like what happened to us today. In Israel, currently, there are no schools. There are limited educational establishments. So juggling these two roles. And on top of that, taking care of an elder family member. So in addition to the routine help, they have to take care of the COVID-19 toll. And there are some papers and articles about this issue. For example, caregiving is never easy. COVID-19 has made it harder. And indeed, some quotations, I worry if I get COVID, there isn't a person who could step in. Another person who interviewed said that my longtime health aides are now unavailable. They may risk her mom. He's not well, but moving him to a senior care facility may risk his life. Every cough makes me worried. And I'm even afraid to hug my kids. I might contract the virus and risk her. So there are a lot of issues here. And it's actually acknowledged by the World Health Organization, the CDC. There are some guidelines there. The American Red Cross. Also in the UK, by the government, there are guidelines, and even in Israel. So it is acknowledged, but still it's not enough. So even before COVID-19, some people we interviewed said that when my mom was hospitalized, I have attempted to cover my absence by working at night. I've tried to be in 100 places at the same time, take care of my children and my mom. Yet my workplace showed intolerance and lack of empathy, and I felt they just waited for me to leave. And we heard it again and again. So we found some gaps in the literature. First of all, there were really few studies that specifically focused on sandwich generation caregivers. There's lack of comparison to other types of caregiving, like those who care for children or elders or non-caregivers. And most studies were cross-sectional, and we don't know how organizations can help. And I'm affiliated with the School of Management, so I'm really interested in what organizations can do. So we found 15 studies that focused on sandwich generation caregivers. They found that they are more likely to develop job burnout, absenteeism, neglecting one's health, sleep problems, marital problems. And what we wanted to know on top of this is, first of all, are they at increased risk for physical and mental deterioration with time? And what can organizations do to mitigate this effect? So the study I'm going to talk about was recently published, and I can email it to you later, The Depressive Price of Being a Sandwich Generation Caregiver. So just a brief look at it. We wanted to know whether caring for both elders and children results in deterioration in physical health that leads to changes in depression and increase in depressive symptoms. And we specifically wanted to compare sandwich generation caregivers to those who care for either children only, elders only, or care for none. We also wanted to know whether support mechanisms mitigate these effects. We looked at both organizational support mechanisms, work-life integration practices like taking a leave or flexible work arrangements, and supervisor emotional support. We followed a cohort of more than 1,000 Israeli employees in 2 waves, in 2002 and then again in 2014. These employees completed surveys while they visited Center for Routine Health Examinations in Israel. So these are routine examinations, they were healthy. We also thought of possible confounders such as carrying, first of all, their characteristics, maybe their age, gender, or socioeconomic status is important. Their occupational characteristics like blue or white collar jobs, job tenure, work hours, managerial position. Other characteristics like the length of caregiving, how many hours a week, how many years, parenting a toddler. So the list of possible confounders was really long. So you won't be able to read this table, of course, but as I said, it has been recently published. But what we did find is, first of all, caregiving status increases the likelihood of experiencing an increase in depressive symptoms. So those who were sandwich generation caregivers did have an increased risk, and it was partially mediated through a deterioration in physical health. We did find that those who cared for children or did not care were at a reduced risk, and the differences between sandwich generation caregivers and those who care for elders were milder. Namely, that what is really important here is understanding that taking care of elder persons, and this was before the COVID-19 pandemic, really takes a toll. As for support mechanisms, so we looked at 2 support mechanisms. First of all, organizational support, like taking a leave of absence, taking an unpaid vacation, or leave an absence to take care of a sick family member. So we did not ask them whether they utilize these mechanisms, but rather are they available for them, because some of them were taking care of family member for 2 or 3 years. So apparently they don't have to take care of them all the time, to utilize them all the time, but at least occasionally. And we saw that the number of practices available by the organizations, the more they increase, the less likely sandwich generation are to experience an increase in depressive symptoms. In fact, they experience a decrease, whereas all others, they did not experience a major change in depressive symptoms. So we saw that it actually helps them reduce the number of symptoms. Again, with managerial support, we looked at willingness to listen to problems, take time to learn about the personal problems, even talk effectively about solutions. And again, we saw a similar trend. The more supportive your supervisor is, the more likely you are to experience a decrease in depressive symptoms. And for others, there was no change. Few limitations. These are self-reports of health, depression, and support, but it is a longitudinal study. And we did not ask who they care for, so maybe we should have differentiated between elder care and other chronic conditions. The culture is, of course, this is the study based in Israel, maybe it had an impact. But if we look at takeaways, first of all, sandwich generation caregivers are at increased risk, and we don't think about them often. We usually think about family work issues and we focus on parents of children. And COVID-19 has increased the demands placed upon them and may, I guess, result in profound physical and mental deterioration. And I think that we should acknowledge that. Second, family supportive organizational practices should not focus on childcare only, and organizations are really advised to offer specific help to caregivers of elders too. There are numerous ways to do that. Managers should also be notified of sandwich generation caregiving challenges, look for symptoms of depressions and offer solutions because there are solutions. And I think that sandwich generation caregivers should be prioritized when allocating mental and physical support resources. So if a manager has to decide whom he should give some days off, I think the sandwich generation caregivers should be first in line. So that's it. If you want to contact me, and I'll be happy to send you the paper. Just look me up on Google. You'll see my website at the College School of Management. Thank you. And again, my apologies.
Sharon, thank you very much for a great talk. Very interesting subject area. Again, if any of you have questions, please use the participant function and then raise hand. Hopefully you can find out. Pressing the 3 dots on the participants box will give you the raise hand function. So maybe I can start by asking about any positives of being a sandwich.
Yeah. So the literature talks about self, fulfillment and meaning in attending others. But as a matter of fact, there aren't a lot of upsides. There are more upsides to caring for children because we see them grow, there's the future, etc. But if we think about caring for elders, usually you don't know how long is it going to take, whereas with children, you know when they're going to, turn 18. It is really unpredictable because oftentimes their health condition may change within a week or day, and you don't know how long it's going to take, when they'll come home. And even financially, just trying to predict how long you should pay for a specific medicine or pay for a healthcare, you don't know that. So the toll is huge. And another problem is that they cannot talk about it with no one. So they cannot complain to their children that they have to take care of the parent, and they cannot complain to the parent that it's difficult. So we've looked for positive things, but it was difficult to find some upsides.
Great. Well, of course, it's not one size fits all. It really depends on the age of the children, of the elderly, health conditions, and so on. So it's very difficult to assess this. I understand that.
Yeah.
Did you look at biomarkers of stress or aging biomarkers in these individuals? Giving
we've actually looked. We had some data on C-reactive protein levels. We found no indication for any association with them. We didn't have other biomarkers. Oh, we did look for biomarkers, not biomarkers, but hypertension, and there was no association. But we should remember that that caring for both children and elders is really a chronic condition. So you talk about 2, 3, 4 years. This was the average in our cohort. So you wouldn't see, like an increase during this time in specific biomarkers. I guess. So when they completed the survey, they were already taking care of the others for several years. So maybe there was a deterioration. We just couldn't see it.
Yeah. Ehud has a question. Maybe if you can mute yourself and ask.
So I wonder whether you looked at the situation in which the elder are suffering from last stages of or advanced stages of dementia, because on one hand it seems to have more burden on the caregivers, but on the other hand, maybe because it's more of like a practical caregiving and not less of emotional or less of, I would say, intellectual. How it affects the health of the caregivers themselves?
So I wouldn't base my answer on my own research because we didn't study that, but I did read a lot about these issues. So people with dementia, it becomes, so usually people with like advanced dementia are put in care facilities, and then like the care have to, you know, suffers less in terms of time, etc. But the mental toll is huge. We had some issues like this in our family as well, and people stop recognizing their children. You see this deterioration, which is terrible, and the mental toll is quite huge. So with the deterioration, we see not me, but others saw an increase in depressive symptoms and yeah, such.
Karen has a question.
I do, thank you. Sorry, I'm not playing by the rules, but it seems that as a host, I can't actually raise my hand in the conventional way. So on again, a positive note, what can we do, or we as in more of a system and administration, to try to get people to change? Is it enough to just send out notices and say, be considerate? And in fact, now we have to be really considerate during the COVID. A lot of people are stuck home with their kids. And so maybe work productivity isn't as high, but there's just nothing you can do. How do we make sure that everyone is showing their sympathy and understanding?
Right. So even on top of just sympathy, I think that many people were not aware that people can use their sick leave to care for others. So there are some, but the word, sorry, there are some practices that organizations should provide their employees with, and both the organizations are not aware of it and the employees are not aware of it. So raising the awareness that this is something that organizations should actually do is important. And this is one thing that we've actually done following this research. We had some publications about it, like in the public media, and people actually wrote that they were not aware that as employers that they should or can help their employees. So raising awareness is not just to the mere, you know, to just being sympathetic, but also to what are their rights, legal rights, and there are legal rights. This is one thing. And the second is raising managers' awareness to the symptoms that these people might suffer from and really ask them to prioritize. I think that this is something important. So if you have, you know, direct employees, some of them care for both and some for children only, you should bear that in mind, because people all the time think about just parenting young kids, whereas these people suffer from other issues, like the sandwich generated caregivers.
Great, thank you. One last quick question, maybe from Mira, and a short answer, please.
Yeah.
Okay, thank you very much. I just wanna, you know, while we are sitting, everybody in this webinar, I wanna just take it to the digital world that we are facing right now, and to the tools that apparently we have to help these people. And my big question that I'm not sure you touched that as well, but just to phrase this interest, how much, or if at all, did you relate to this digital help, especially between the kids and the elderly, each one by itself, and maybe like was done in England, even to combine them and maybe go mentally, but not only, also physically to help that?
So the thing is that this study was conducted before the COVID-19 pandemic. So, and it was the second wave was in 2014. So we didn't have these digital facilities yet. I guess they might be helpful. There's even a program now. The Israeli government should or supposed to hand out laptops to or iPads to elders, and this may help. But what we've talked about is really physical help. So, you know, groceries and taking them to examinations, etc. So it's not just the mental connection.
But can't you combine? Isn't that a way?
I guess you can.
At all?
We haven't studied that. It was, you know, several years ago, so it was less available, I guess.
Great, thank you very much, Sharon. Because of time, we'll move on to our final speaker, but that was a great talk. Thank you. So our final speaker this morning and for today is Heidi Johansen-Berg from Department of Clinical Neuroscience at the University of Oxford. She's the director of the Oxford Centre of Functional MRI of the Brain and the Wellcome Centre for Integrative Neuroimaging. So maybe if I can invite Heidi to share her screen. And her talk is about the microstructural MRI of the aging brain. Thank you, Heidi.
Thank you very much. So hopefully you can see my screen now and you can hear me okay. So thanks very much for the invitation to join you today. It's been a really interesting meeting so far. I'm going to tell you about a new project. This is a new collaboration, a newly funded project, which brings together the University of Oxford in the UK with Tel Aviv University in Israel, and it involves myself, my colleague Claire Mackay, and Yaniv Asaf from Tel Aviv. And our project is aiming to look at brain imaging in the context of aging. And really it brings together a number of different areas of expertise, which both centres have, but to differing degrees. And by working together on this project, we can really complement one another's experience and skills. So we in Oxford, my group have been interested for some years in the relationships between lifestyle factors and brain aging. So to what extent do our lifestyle choices influence brain health and brain aging in later life? And we've also been particularly involved in Oxford in so-called big data neuroimaging, so projects where brain scans are acquired in large numbers of individuals so that we can leverage that big data to try to understand variability, so why some people age more healthily than others. And then Yaniv particularly brings expertise in advanced brain imaging methodology, so really pushing the innovation, particularly in terms of the acquisition and analysis of really cutting-edge brain MRI techniques. And I'll talk you through how those things will hopefully come back together in this new project. So this is just a flavour of the types of headlines that I'm sure you will all have seen. These are from UK newspapers. I'm sure you have the same. Also in Israel, of all of the different kinds of lifestyle choices and activities which we're told can either make us age better or worse. So just some examples here is exercise, sleep, puzzles, learning a language, etc. So there seems to be a new article each week on ways in which we can optimize our brain aging. So, but if we wanted to study this systematically or quantitatively, we can expect that the way in which each of us will age will depend on a number of different characteristics, which vary in how modifiable they might be. So to some extent, we know that it will be determined by our genetics, over which we have no control, and hereditary factors. We know that there are certain influences of our environment, over which we have minimal control, and then our own personal life habits. And arguably that's where we as individuals have the most control. But as you've seen from that flavour of headlines, the message that we can get from life habits can be a bit confusing. So eat healthily, don't eat fat, eat good fat, don't eat carbohydrates, don't drink a little bit, don't smoke, don't use cannabis. Cannabis might be good if you're older, do sports, don't overdo sports, etc. We can get mixed messages from those snapshots looking at relationships between individual lifestyle factors and brain health. Ideally, what we would want to come up with is individualized recommendations based on monitoring lifelong effects of our lifestyle choices on our brain physiology, and the kind of data that could help us move towards these sort of individualized, personalized recommendations might be these big data brain imaging studies of the type that I mentioned, where we can leverage large data sets to understand variability and to make individual subject level predictions. So just to illustrate that schematically, if we really understood the trajectory of different brain measures over the lifespan, particularly over later life, then you can imagine looking at somebody in middle age, determining where they sort of sit on this trajectory, and then giving that person individualized lifestyle recommendations to try to take them sort of above that line to make them age more optimally given their own starting point. So in order to determine what these trajectories are and to determine what the associations are between our lifestyle choices and our brain aging trajectories, what we need are these large data sets which quantify that type of variability. And there are many, many international projects which are aiming to collate those types of large data sets. So these are just a few examples across the world, from the US, from the UK, China, etc., where large numbers of individuals, often including later life individuals, have had a number of different types of brain scans, as well as measures of their health and lifestyle. And some of these data sets have been made openly available to the scientific community. And so people are starting to mine these data sets to try to understand the relationships between lifestyle choices and brain aging. These data sets vary as to what information is contained within them, what types of brain scans are contained within them. All of them would contain these sort of conventional structural brain scans, so-called T1 weighted scans of the type that you might get in a clinical setting. And often we would analyze those scans then quantitatively across a large number of individuals using different types of techniques, either to measure the volume of very specific structures, such as here, part of the hippocampus, or to look across the whole brain volume to use so-called voxel-wise techniques to try to understand which areas are bigger or smaller, or shrink more or shrink less in different populations. Some of them might also include other types of scans. So a commonly included scan would be a diffusion imaging scan, which is particularly useful for giving us information about the structure of white matter fibers. It can tell us about the structural integrity of white matter fiber bundles, for example. This is one example image here where we're plotting a parameter called fractional anisotropy, which tells us something about the structural integrity of the white matter. So these are these sort of fairly conventional measures of brain structure, which would be included in many of those large data sets that I described to you. And so people have looked at variation in these types of brain measures in relation to different lifestyle factors. And I'll just talk you through a few examples from my own group where we've looked particularly at sleep and physical activity in later life. And just to give you a flavour of the types of findings that we've observed by looking for cross-sectional associations between one of those brain measures and a measure of lifestyle. And this will all be work done by Claire Sexton and Nayara Demnitz in my lab. And so the first example up here, in a collaboration with Oslo, we were able to show that people who slept poorly, so poor sleep, was associated with greater brain atrophy. So by scanning the same individuals over time, we can quantify how their brain atrophies or shrinks over time. And we found that in older adults, shown by the blue line here, the worse their sleep quality, the greater the decrease in brain volume over time. So the worse we sleep, the more our brain atrophies over time, particularly in later life. Poor sleep was also found to be associated with worse white matter structure. So using that diffusion scan that I mentioned, we found that particularly in the frontal parts of the brain, there were poorer measures of white matter structure in individuals who slept less well. Physical characteristics also were related to brain aging, so mobility. So for example, our ability to get up out of a chair, to walk, to balance on one leg. All of those were associated with the size of the grey matter in the cortex of the brain in different areas in older adults. And then physical activity, so the degree to which we get active during the week over middle age, was associated with white matter structure in this example here is from the Whitehall cohort in the UK, where people who have been followed up over many decades from middle age to late life, those who were sort of consistently inactive over middle to late to older age had worse structure in the white matter in very diffuse areas of the brain. So this just gives you a flavour of the kind of associations we can find by looking at one of these lifestyle factors, physical activity, for example, in relation to one of these types of brain scan. One thing that we want to do differently in our new project is to make use of new imaging methodologies, both to acquire and to analyse richer types of data than has been possible to date. So thinking first of all from the analysis point of view, these are all what we call unimodal associations. So one brain measure associated with one lifestyle measure. We expect that we can improve our sensitivity to relationships between brain and aging through so-called multimodal measures. So this is where we combine lots of different brain imaging metrics together to try to understand what combination of brain measures best relates to brain aging. So I'm not going to go into this in any detail at all, but just to give you the idea, we have different kinds of brain scans, each of which we can parameterize in different ways. So we have a score for each of our brain voxels. Each of those parameters is capturing a slightly different aspect of the structure of the brain in these cases. Conventionally, we would look at them all separately, but we can combine them all into modes of variation where we can generate maps which tell us that some combination of those different brain measures relates to variation in brain aging, for example. And just to illustrate that approach, I'm going to show you a study done by my colleague Gwenel Douard in Oxford, who, again in collaboration with Oslo, took a lifespan data set. So this is brain scans in subjects from 8 to 85 with a number of different brain structural modalities, in which she was able to feed in those different structural modalities using this type of methodology to ask, show me maps of the brain which describe inter-individual variation over age in this case. So spatial components that characterize age-related variation in brain structure. What she found, unsurprisingly, is that by far the most dominant component is this monotonic decrease with age. So this is not surprising. Over the whole of the lifespan, pretty much the whole of the grey matter of the brain effectively shrinks as we get older. What was interesting, though, is because this is a completely model-free approach, a data-driven approach, where we're looking for any kinds of patterns in the data, anything meaningful that describes variability between people, we're not only limited to seeing the things that we expect to see, but we can also reveal less expected patterns. And the next most prominent mode of variation was this inverted U-shaped relationship here. So you can see that for this brain map, whose spatial distribution is shown in the brain image here, we see this inverted U pattern, which you can think of as a sort of last in, first out sort of pattern, whereby aging mirrors development. So thinking about how to relate these different patterns to age, what this deconstruction of the data is showing us is that areas of the brain that sit within this spatial map here, their variation over the age span can be explained by a combination of these two different components. So we can plot that then. So that means that for the areas inside this map, compared to the rest of the brain, these areas develop more slowly, but then degenerate more quickly. So that sort of captures this kind of last in, first out pattern, such that areas within these within this map show this protracted brain development. They develop more slowly, but then when you get to later life, they degenerate more quickly. And this was particularly interesting because Gwen did a number of comparisons between the spatial distribution of this pattern in a healthy aging lifespan with a number of different diseases of aging or disorders of development. And what she found was that the spatial pattern of that age-related inverted U shape that you see here in yellow in these plots was remarkably consistent spatially with patterns that had previously been shown to be associated with accelerated atrophy in Alzheimer's disease, shown here in blue, or with altered development in adolescent onset schizophrenia, shown here in green. So obviously completely different disorders with totally different underlying pathology, yet there seems to be something especially vulnerable about particular brain areas which might reflect their developmental and age-related pattern, even in the absence of disease. So perhaps these areas that are late to develop and then quick to decline in aging also have particular vulnerability to disorders of development and aging. So that was really just to illustrate that one way in which we might increase the richness of our brain imaging data is to combine across different types of brain scans. We can also use multivariate analysis methods again in a data-driven way to look for associations between combinations of brain measures and combinations of lifestyle measures. So again, just to illustrate this approach now in a developmental project, we can use methods such as canonical correlation analysis to look for multivariate patterns. So again, we're looking for modes of variation, but now between combinations of brain measures, combinations of physical lifestyle measures in this case. And just to illustrate that in the adolescent brain, we found across a group of young adolescents that those with a more active lifestyle, characterized by a combination of these physical measures, have particular patterns of brain structure and blood flow. So this figure at the bottom here illustrates this mode of covariation which comes out of this data-driven analysis, such that these patterns of brain measures, so in other words, areas with more diffusion, more perfusion, sorry, this is vascular perfusion, and faster arrival time of blood and greater volume in certain areas of the brain are associated. So this is the brain side of our relationship. This is the physical side of our relationship over on the right here. You can see that those perfusion and brain structural measures are associated with higher fitness, more activity, lower body mass index, and lower periods of sedentary activity. So again, this is just to illustrate how leveraging these data-driven multivariate approaches can give you greater sensitivity to the true underlying richness of the relationship between lifestyle and brain. So the other way in which we can push the boundary of what we're doing is to try to push the resolution of our brain imaging techniques. So conventional, our brain scans are worlds apart from what we're truly interested in, which is often at the cellular level. So this is giving you an idea of the sorts of resolutions. We're typically imaging, say, 1 millimeter isotropic voxels in the brain, which obviously will contain many thousands of brain cells. So you can think of us being very much zoomed out compared to the level that we might actually be interested in. And this is really where the Tel Aviv group comes in, where Yaniv Asaf and his colleagues have developed some really exciting new technologies over the last few years that allow us to image getting closer to information about things at the level of cells, not necessarily by acquiring things at higher spatial resolution, but rather by using very, very clever physics and modeling to be able to extract cellular level information even from our relatively crude measurements. So just to illustrate the kinds of methodologies that they've developed, this is a technique called Excalibur, which uses diffusion signals, but modeled in a way that allows for estimating of axon diameter distributions. So we can get a sense of the diameter distributions of axons in the white matter of the brain. This is a very nice new technique that they have developed to allow for imaging of cortical layers. So based on the difference in T1 weighted contrast between the cytoarchitectonic layers of the cortex, which might, for example, be driven by difference in myelination between different cortical layers, they're able to resolve that layer structure in the cortex non-invasively. And all of this can be brought together to derive so-called connectomes, so wiring diagrams of the human brain based on this structural information, looking at things like fiber tracking and axon diameter distributions, and then combining that with resting state functional MRI data to derive functional connectomes. So we can combine all of this information along with functional imaging to derive much, much richer connectomics, which tell us about connectivity at the human brain at the level of microstructure. And that's exactly what we're hoping to do with the new project, which should be starting this month, where we're aiming over the next couple of years to scan 500 subjects across the 2 sites, using those novel microstructural acquisitions, as well as acquiring a whole bunch of non-imaging measures, including lifestyle measures. So the outcomes of that then will be to provide this first microstructural connectome and information about its variation across age. That will become an openly shared database that will be shared with the community, and we'll be able to mine that database to detect these novel relationships, multivariate relationships between combinations of brain measures and lifestyle and other factors. And that's it. Thank you very much for your attention, and I'm happy to take questions.
Heidi, thank you very much. Wonderful talk. Very interesting. Please do use the participants and raise hand function if you would like to ask Heidi a question. Maybe I can start by considering a comment rather than a question about regional differences. That means global regions. It's not one size fits all. How about different ethnic groups, different environments, and so on?
Yeah, it's a very, very good question. And most of the big data sets that I referred to are not very rich in that regard. So some do contain it. Some have made an effort to try to sample their populations more representatively in terms of ethnicity and to capture some of that information. But obviously they are to date all from a particular geographical region. Many of them aren't very representative even of the population, the geographical location that they are. So I think that's one of a number of variables that isn't very well represented or captured in the data that are there at the moment. And I think one of the motivations for Yaniv in proposing the current project is, for example, to try to acquire some of these types of data in the Israeli context, whereas I understand until now there hasn't been this type of big biobank type database, including brain imaging measures. So that's something that he hopes to be able to better understand for that location and population, how some of these different variables could relate.
Great. There's a question from Robin Franklin. If Robin can unmute you and ask the question.
Yeah, hi Heidi.
Hi.
Hi, that was a terrific talk, and the plans you have are really very exciting. I have a question about the connection between brain atrophy and sleep, or sleep deprivation. And this may be speculation, or we may know the answer, but I mean, what's the sort of mechanistic connection between those two observations? And it's difficult for me in my rather limited way to think what they might be, but it's intriguing.
Yeah, I mean, obviously with any of these cross-sectional associations, we can't determine causality. So there could be all sorts of other things which are influencing sleep, or there could be all sorts of other ways in which poor sleep, influences mood, which then impacts negatively on aging. So I think it's obviously very hard to disentangle causality. However, I have obviously thought about that, and I think there's various different possible mechanisms which could mean that was a causal relationship. One of them being myelin, which, as I'm sure you know, there is really intriguing evidence about differences in oligodendrocyte dynamics during different phases of sleep. So that's a particular mechanism that I'm interested in looking into, whether to what extent disruption of that usual oligodendrocyte cycling over sleep could then have long-term consequences, but whether that explains this particular relationship, I don't know.
Great, thanks.
Another question by Ehud.
Yeah, thanks Heidi for a great talk. I follow up of Robin's question. How do you measure the quality of sleep?
Yeah, so good. In that particular example, it was purely questionnaire based. So with many of these large scale projects, you are limited in the sensitivity of the measures. So that was just a standard Pittsburgh sleep questionnaire, which is a standardized scale with a sort of cutoff for good and bad sleep, as well as a continuous measure. In some of the slightly smaller data sets, we have things like actigraphy, which provides an objective measure of activity from which you can infer sleep quality. Again, there's lots of questions over how to do that optimally. You know, ideally we would have, say, sleep EEG, but that's very difficult to do at scale.
Great, thank you. Any other questions? I know we're running slightly a few moments over time now, but if there are no more questions, just leaves me to thank again the speakers of this session, Ehud, Sharon, Dina, and Heidi, for great talks. Thank you for keeping to time, and thank you to the audience for the discussion and asking questions. So we end today's session here. I'll hand over to Karen in a moment, but I'd just like to again thank the organizers at Tel Aviv University, British Council Israel, of course, and my friends there for putting together this virtual event. And I look forward to seeing you in person next year. Thank you, Karen. Over to you.
Hi, Richard. Thank you. So I can only echo your words. And I think that 4 minutes is still pretty good. Everyone was really wonderful in keeping to time so that those of you in the UK can go have lunch and, I don't know, a late afternoon snack for the Israelis, and for the rest of you, whatever you'd like. So we will be releasing the recording of this day, and there'll also be a written transcript for those who will find that helpful. And I look forward to seeing all of you tomorrow, 9 a.m. UK time, 11 a.m. Tel Aviv time. Thank you again very much, and good night.